welcome to the second day of BioStock Life Science Summit. Today we are joined by Targinta and the CEO Per Norlén. So welcome, the floor is yours.
Thank you so much. So Targinta is an oncology biotech company developing targeted antibody therapies for aggressive cancer. Today Targinta is a subsidiary of the stem cell company Xintela, but a process has been initiated to spin out Targinta as an independent entity. So this presentation is a first flavor of that independent Targinta. So with that I will move to some highlights. Targinta develops two products, two first-in-class antibodies, both targeting aggressive cancer, triple-negative breast cancer and glioblastoma. And both are targeting our novel and unique oncology cancer cell marker, integrin alpha-10 beta-1. We have also a robust patent portfolio covering the use of alpha-10, such alpha-10 antibodies for the treatment of aggressive cancer. The business model of Targinta is rather simple. We focus on the early stages of drug development, essentially preclinical drug development, with the intention to out-license or find a partner before starting clinical trials in cancer patients. So what are these lead cancer indications? We focus on triple-negative breast cancer and on the brain cancer glioblastoma. These are two very aggressive cancers with extremely poor survival. Still, it's large cancer populations, and today there are essentially no effective drugs out there. So in a summary, you could say there is a large market potential and a huge medical need. But why would Targinta's products be effective? That comes down to the target, to integrin alpha-10 beta-1. One factor is the expression, high expression in aggressive cancers and in triple-negative breast cancer and glioblastoma in particular. You can see some examples in the slides in this section. If we look at the target in more detail, it's called integrin alpha-10 beta-1. It's part of the integrin family. Integrins in general are cell surface receptors that allow cells to attach to the surroundings. And that's really important for their survival and ability to migrate. In the context of cancer, that means it's important for the ability of a tumor to grow and to metastasize. Integrin alpha-10 beta-1 is particularly interesting because it's highly expressed on a lot of aggressive cancers. It's expressed on the cell surface of such cancer cells, and you can see a beautiful example on the right-hand side. Alpha-10 beta-1 glows on the surface of such cancer cells in this slide. And this is really important, for at the same time there is no or virtually no expression in normal cells, which allows for a very selective and targeted treatment of such cancers. Validation is extensive today. We have, and I showed you just before, and here's other slides on breast cancer and glioblastoma. What you see in dark brown is expression of alpha-10 beta-1, integrin alpha-10 beta-1 in such cancers. So we have validation in triple-negative breast cancer and glioblastoma, but also in other indications such as prostate and pancreatic cancer. And there is external validation in other cancers like melanoma, lymphoma and sarcoma. So let's look at the products. We have two products today. We have Targ10. We have recently selected a lead candidate, and this is a function-blocking monoclonal antibody that is developed to inhibit tumor growth and tumor metastasis. And this product has just moved from preclinical research to preclinical development, essentially meaning that we now will develop a preclinical regulatory package for this specific compound in order to take it to clinical trials in the future. The other program is Targ-XX. That is a program where we intend to develop an ADC, an antibody drug conjugate, which is an antibody armed with a highly potent chemotherapy toxin. This is a product where we don't yet have selected an lead, but that's about to come quite soon. I will start with giving you more information on TARG10. So this is a function-blocking integrin alpha-10 antibody. And this receptor allows for selective targeting of cancers expressing integrin alpha-10 beta-1. We have already shown that we can suppress cancer cell proliferation and migration, and we've also shown that we can reduce tumor growth, both in glioblastoma and triple-negative breast cancer. And there is a quite remarkable graph shown on the right-hand side, where you look at the growth of glioblastoma over time, and in gray you can see the growth of the tumor after treatment with a control antibody, and in blue you can see the greatly reduced growth after treatment with an integrin alpha-10 antibody. Integrins are especially important for migration of cancer cells, and by that they're also important for metastasis. And we have also shown now that our function blocking antibody can reduce and inhibit metastasis in such models. To the timelines. TARG10, we have selected a lead candidate. On that we have also demonstrated proof of concept, and we are now initiating preclinical development. Next year there is a lot of important milestones. We will start cell line development. That means initiation of the first part of production of the product for future clinical trials. We will also conduct safety assessment and start generating a robust preclinical data package for future clinical trials, which are set to commence in 2024. Before that, we will try to identify and find a partner either through out-licensing or partnership. In parallel, we are developing the TARG-XX program in order to generate an antibody-drug conjugate against the integrin alpha-10 beta-1. On the right-hand side, you can see a picture of what that is, an antibody-drug conjugate. In grayish blue, you have the antibody, and attached to the antibody are some highly potent toxins, looking like green stars. We have now evaluated a number of toxins and antibodies and have generated these antibody conjugates, and right now we are evaluating these for both efficacy and for safety with the intent to select a lead candidate. So let's look at some of the data. First, we have confirmed that this product can induce programmed cell death. And if you look on the right-hand side, it's quite impressive. Very strong induction of programmed cell death in a tumor model when you treat with an integrin alpha-10 antibody drug conjugate, but no effect whatsoever if we compare it to a control antibody drug conjugate. Also, we have shown efficient internalization of the ADC. That essentially means that once the product binds to integrin alpha-10 on the surface of the cell, it will be rapidly taken up into the cell, release the toxin inside the cancer cell, and kill the cancer cell from the inside. We have also demonstrated a reduction in tumor growth, and with that we are now quite confident that we will be able to select a lead candidate early next year in Q1 2022. So with that I will move to summary. In Q4 we will now start the preclinical development of TARG10, our recently identified lead candidate. Also, we will spin out Targinta as an independent company, and the process has been initiated, and we will tell you more about that quite soon. Early next year, TARG-XX, and we look forward to identify a drug candidate for that product and program. We also look forward to change the name to something that you can say, and we will come back with that in Q1. Also, we will next year in the spring initiate cell line development, which is the first part of production for either one or both of these products. So with that, I'd like to thank you and for this introduction of the emergence of Targinta as a new oncology biotech player. Thank you so much.
Thank you for an interesting presentation.
Thank you.
So which of your previous experiences will be particularly useful in the role as CEO of Targinta?
I think my experience is very relevant for the role in Targinta. I come with a lot of experience from leading the development of anti-cancer antibodies from discovery, from ID, all the way through to clinical trials. Also leading the transition of a company with a very strong research focus to a drug development company in a public setting. That will become useful.
And can you say something more about the spin-out of Targinta from Xintela?
Yes, I can say that the process has been initiated and we will spin out Targinta as an independent company. The details will be communicated, but there are still some decisions to be formally taken, so we'll come back with that very soon.
And how do you view the potential for Targinta's cancer therapies?
I think it's great. We have a unique oncology target with functional importance in aggressive cancer. We have a robust patent protection and high quality products. So there is a great potential to improve survival for cancer patients.
Yeah, and we look forward to following Targinta's journey. And thank you for coming here today, Per.
Yeah, thank you for having me.
With us now we have a special guest, a market expert, Karsten Boring, who is head of listings and capital markets at Nasdaq Copenhagen. Karsten, thank you so much for joining us today.
You're welcome, and I'm happy to be here. It's always a thrill to be with BioStock.
I wanted to start by asking you, looking forward, where do you think the IPO trend is going from here, say in the upcoming 6 to 12 months?
Nasdaq has welcomed, I think, more than 140 companies during the first 3 quarters here in 2021. And it is really a record year. And if you look back and you see year to date at last year, it was only 44 listings. So it has been a tremendous IPO year, both in numbers of IPOs, but also in capital raised at our Nasdaq Nordic markets. So it is a record. And during the 3rd quarter alone, Nasdaq in the Nordics welcomed more than 30 new listings, raising more than 1.1 billion euros in new capital for these companies. So if we look a little bit further, and after this record-breaking half year and the 3rd quarter where the momentum has carried on, the 4th quarter pipeline looks really, really promising. And also if we look into to Q1 and Q2, because right now we are all the guys here in the Nordic region with Håkan and Niklas, Adam and Erja Mina in Sweden, and Peter and I here in Copenhagen, we are starting to building up the Q1 and Q2 pipeline for new IPOs, and we don't see any slowdown in the pace of companies doing. So it will be possible for many new companies to enter our market. But that being said, we have seen the last couple of weeks with a little bit of higher volatility in the share market than we're used to. And because a lot of investors are not used to volatility, some retail investors has been a little bit nervous the last month or so, but it seems now that we are more into normal water with lower volatility and back on the track with all the new IPOs coming up. So I see only positive things going forward, and to companies that are looking for an IPO during Q1 or Q2 next year, you need to be ready.
In terms of the investor sentiment, which type of companies, and specifically life science companies, would you say have had the easiest time to attract attention from the investors and the capital they need?
Yeah, it's also always a good question to see if there are some sectors that are more hot than others. And this year we have listed more than 20 healthcare companies, all in different company with different company profiles within biotech. But most of them have been early stage, so it's difficult to see if it's a special sector or it's just the company people behind the companies that are attracting the new capital. But with overflow of capital in the market, investors are willing to tap into the higher risk that we see in the biotech companies than a few years ago. So it is really good time. So, but if I should pinpoint some hot companies within healthcare, we see a lot of interest around medtech companies, and that's also what we see in the private market. So I will guess that we will see more medtech companies coming into the biotech sector over the next half year.
The Nordic and Swedish stock markets have really active retail investor community, and which plays a very important role in providing capital to early stage innovation companies. And do you think this model, this Scandinavian model, will continue to be successful going forward, or do you see any challenges to this type of financing model?
I really hope it, because it's really a unique model. And it is correct when we look at, especially First North at Nasdaq, we see retail investors to play a unique and important role in the Scandinavian ecosystem for risk capital. And that has only been picking up the last couple of years. So if we looked a little bit about on the shareholder mix before COVID, and here the retail investors counted for almost 80% of the ownership in these small companies. And during the pandemic, retail investors have been even more active. So my best guess is still a high number with the retail investors. And I don't see any problem for retailers continue this journey as long as the companies continues to deliver on the value triggers as they have promised. And then I'm absolutely sure that the investors will be there. For the companies to continue to be in the eyes of the retail investors, biotech companies, they will have to deliver on the value triggers to raise more capital, because they need a lot of more capital to fulfill their dreams. So you have to make it to the market and do a home run for your biotech products. You need really to deliver on your value triggers to keep the retail investors in the companies and to raise more capital.
Next up from our studio here in Stockholm, we have Modus Therapeutics, and presenting is CEO Jan Öd. Welcome, Jan.
Thank you, Mike. And thank you everybody for having me and for your time. So let's talk about Modus Therapeutics and our trajectory towards developing a therapy to treat sepsis and septic shock. Modus Therapeutics is a small biotech company that develops its proprietary heparin-like molecule cevuparin with a focus on sepsis and systemic inflammation. The unique aspect of cevuparin is that it has been modified not to display the bleeding risk and the anticoagulation effect seen with the normal heparinoids. And this is what allows us to target higher doses that also can then explore effects in other important diseases that are not anticoagulation dependent. So what we have found in our preclinical models is that cevuparin protects the blood vessels during septic inflammation. And in order to go further and realize development in patients with sepsis, Modus recently concluded an oversubscribed IPO on Nasdaq First North Growth Markets. The IPO proceeds are already at work to start our first study in this program, a phase 1b LPS challenge study. This is a sepsis mechanistic study, planned to start in Q4 this year or Q1, early Q1 next year. And this will then pave the way for the first patient study, a phase 2a study that is planned to start Q3, Q4 2022. So this is our team. It's a very seasoned team in terms of experience. We have representation from very experienced VC capitalists and big pharma executives. And then, of course, myself with a physician and clinical development background since long in different companies, and also our CFO, Claes Lindblad, with more than 20 years of experience from all aspects of the market and the economic control of life science companies. We also have a scientific advisory board that has been involved in the whole planning and setup of this new indication for cevuparin, sepsis and septic shock. Cevuparin is a unique molecule that is based on heparin, but that has been targeted for modification chemically by modifying a couple of residues, 3 residues here. We go from the pentasaccharide that is typical for heparin and that gives the anticoagulation effects to a disaccharide repeat. This renders a patentability that gives us a patent that extends up to 2036, 2037 with extensions. And the modifications make it possible to dose cevuparin in significantly higher doses compared to normal heparins. This is important because there's a broad set of mechanisms that can be targeted with relevance for other kinds of disorders than anticoagulation. And that is where our indication, sepsis and septic shock, comes in. So a number of different targets have been identified that are potently affected by a heparinoid. And we can target these in a much better way thanks to the modified molecular scaffold that constitutes cevuparin. There is also another advantage. So we have finalized all the early development in terms of toxicology and phase 1 studies. We also have an extensive safety profile in man thanks to earlier development endeavors in sickle cell disease. So in all essence, we can then say that we are ready for phase 2 in sepsis and septic shock development. So looking a little bit closer on the mechanism of action that we have identified in this disorder, we have found that the effects of cevuparin helps protect the inside of the vessels during septic inflammation and systemic inflammation. And in brief, it means that normally in the bloodstream we have a calm situation where inflammatory cells are taking care of the monitoring of any immune threats, whilst at some occasions when bacteria comes into the bloodstream, this can lead to overactivation of these inflammatory cells. These activated cells then, in order to try to fight the threat that the bacteria is constituting, try to do so in some aspect by degranulating and releasing substances that are harmful for bacteria, but they are also harmful for the lining of the blood vessel. And this leads to a chain of events that eventually puts a patient to in a very poor spot, where organ failure ensues from the endothelia opening up and letting out plasma, so we have plasma leakage. What cevuparin does, and what we have seen in our models, is that it can bind and neutralize many of these factors that are important for the damage to the endothelia in this early part of the reaction. So stopping the furthering of the worsening seen later. And just in brief, we can see that, for example, in the lung swelling in mice that are subjected to systemic inflammation, using cevuparin, that lung swelling is taken back to normal, and also looking at cells that are endothelial cells. These cells open up on challenge by bacteria and white blood cells, but cevuparin can put that back to a more normal state. And also here we have characterized the different targets that are constituted by these harmful agents and to what extent they are bound by cevuparin and neutralized. Sepsis is a part of a bigger problem. It's the systemic inflammation disorders, so to say. And this is, just as with sepsis, this is where an inflammatory reaction becomes overdetermined and threatens the patient's well-being beyond what is clinically manageable often. And it can be caused by not only bacteria but also other big trauma, such as extensive surgery and extensive autoinflammation, autoimmunity. And as you can see, there are almost 17 million visits to the US ERs per year that go with some type of systemic inflammation, and sepsis is calculated somewhere between 2 and 3 million of these. And then, of course, the most severe form of sepsis, septic shock, is around 700,000 per year. And this is a very severe situation because the mortality is still around 30% here, and we don't have any specific treatments to use in this situation. So, starting out then with the clinical development to aim for this condition, we're planning to start in Q4 this year or early Q1 next year, a phase 1b study where we provoke a sepsis-like reaction in healthy volunteers. This allows us to study the effects of cevuparin in this kind of systemic inflammation in a very controlled way, which will then serve to inform the important next study in septic patients. And this study then is planned to start Q3, Q4 2022, and to run for the subsequent year after that. This plan also coincides with our financing for the next study, for the phase 2A study. Right now we are working off the capital of the IPO, and within the IPO there were also warrants, and these warrants will vest and strike in a period that then is appropriate to finance the subsequent development. So looking at the treatment potential, if we follow the trajectory of a sepsis patient from coming into the ER, there are a number of different endpoints that we can study. We can study, for example, the symptom and the difference of in symptom parameters between treated and untreated. We can also study the time to worsening or the time to recovery for patients. We can also look at the complications and death rate at day 28, so it's a fairly short treatment period, and also how long patients are in mechanical ventilation, which is a very important cost-containing endpoint. The development that we have discussed here is this part of development. Of course, later then there will be the need for phase 2B and phase 3 studies. And in our business model, we focus on partnering for this kind of indication. We think that brings value faster to our project. And for that, we need to present with potential inflection points. And we believe we have inflection points that drive value, both after the phase 1B study, but more significantly after the first patient study, and potentially we may need to also do a phase 2B study, after which there is, of course, also the potential for inflection and partnering. Modus is also continuously evaluating collaborations with value-enhancing potential. So a good example of that is, while not affecting our cost footprint so much, we're collaborating with Imperial College London on a severe malaria project in patients, and this whole project is financed by the Wellcome Foundation. So, to summarize, Modus Therapeutics is focused on its clinical program in sepsis and septic shock with its unique proprietary drug, cevuparin. So we have preclinical data that indicate a protective role for cevuparin on the vasculature during septic inflammation and systemic inflammation. We're phase 2 ready. The toxicology and safety packages are all in place, and we also have extensive safety data in patient groups and in man. This allows us a fast-to-market approach with clinical study already on track to start by the end of this year or early next year. And also this means that we have an early start to value development. Sepsis and septic shock is a leading cause of death in ERs and ICUs worldwide. And this means that with the market potential of around 6 million for septic shock market, that a successful treatment in this area would have a $1 billion commercial potential. We also have multiple shots on goal with further indications within the systemic inflammation area. As you remember, sepsis is a part of a bigger spectrum of disorders within systemic inflammation, and there could be possibilities, having a sepsis and septic shock treatment that works to also broaden into other types of systemic inflammations. We're also looking at patent protection up until 2036-37, and we're also conducting work that will have potential for new IP in pipeline. Thank you for listening. Thank you for your time.
Thank you so much for that presentation, Jon. I'd like to begin by asking you, in general, sepsis, what makes it so difficult to treat?
I think there's an easy and a difficult or a more complicated answer to that question. But I think there are a couple of basic factors. When we talk about treating sepsis, we actually talk about treating our own immune system, because that is what is driving the reaction the over-determined reaction of our immune system towards ourselves. So it's us fighting ourselves, because that makes it difficult. Another aspect is, of course, that it's a severe condition. So once we start treating it, patients may already be quite in a quite bad place, this means that, you know, this is related to another problem, and that is identifying the patients at an early stage enough to be able to have treatments that work. And the third step is, of course, also we don't have any specific treatments yet. So the treatments and standards of care that are used are general intensive care drugs. They are used in a good way, but I believe that in order to, you know, drive death numbers and complication rates down, we need to see more specifically approved medications coming onto the market and being employed at the correct time for these patients.
Absolutely. So, well, you just said that no other real treatments exist in that sense. How does Cevuparin compare to other treatments in development, let's say?
Yeah, so I think a unique part of what Cevuparin does is that we target a number of different harmful agents that participate in this reaction that harms the blood vessels from the inside. And I think we differ from most competition in that sense, because most other compounds in development have a more narrow scope, focusing on one molecule, one mediator, one cytokine of the septic reaction, or a specific organ, the lungs or the kidney. Whereas the effect of a heparinoid or a glycosaminoglycan here is to act in the whole systemic compartment, so to say, all of the blood, with this broad effect.
Okay, so much broader effect in general terms. So you mentioned the plan is to go to the clinic with Cevuparin. Could you tell us a little bit about how the clinical studies will be set up?
Yeah, so as I briefly showed in the presentation, we choose to start with a phase 1b study, which is not really a phase 1 study in its proper sense. Modus has concluded all the necessary regulatory phase 1 studies in order to go into patients. This special phase 1b study is a situation where we provoke a sepsis-like situation in healthy volunteers. That's why it's still called a phase 1b. So an LPS challenge study is where, in our case, we provoke both the skin very locally to simulate a local tissue inflammation, similar to what happens out in any tissue, with a small dose of LPS, with and without Cevuparin in different doses, and also where we give a larger quantity of LPS into the bloodstream of the patients, and then they really will experience a early reaction similar to the early phase of a septic or systemic inflammation. And then we also study the different doses of Cevuparin here. We hope that we will be able to extract very important observations here that will facilitate and make the next patient study more efficient and effective. Because in the real world, in the septic patient situation, things can be quite chaotic. And if we have important information such as dose level and indicators of when to start treatment and so on, that would be very valuable to trigger the patient study after.
Absolutely. Well, great. Well, thanks so much for answering the questions and for the nice schematic. It'll be helpful for our viewers for sure. And thanks for coming here today. We wish you all the best.
Thank you, Mike, and thank you everybody for your time and for having me here.
next presenting company will be Respiratorius, and with us we have CEO Johan Rott. So welcome, Johan, the floor is yours.
Thank you very much for the introduction. Respiratorius was founded as a research spin-out from Lund University in the late 90s. The initial focus of Respiratorius was to develop new effective candidate drugs for treatment of respiratory diseases such as COPD and severe asthma, which was then a therapeutic area lacking of effective drugs. In year 2012, Respiratorius acquired the oncology asset VL-001, which has then been developed through clinical studies and is now being prepared for a phase 3 study based on the very positive results from the initial studies. The new candidate drug for COPD and asthma is RCD-405 and is now being prepared for a phase 1 study. The business model of Respiratorius is built on out-licensing of the projects. And we are actively seeking partners for the late-stage clinical development and commercialization of the projects. We have ongoing partnership discussions, although much attention has been towards completing the product VL-001 and initiating the PK study. Going a little bit deeper into VL-001, it is for the treatment, and it's a new treatment for diffuse large B-cell lymphoma, which is the most common type of non-Hodgkin's lymphoma. And annually about 60,000 people are diagnosed with diffuse large B-cell lymphoma in US and Europe. The overall 5-year survival is approximately 70%. But the problem is that 10 to 15% are primary refractory to the first-line therapy. And 40% of those who actually respond to the first-line therapy relapses within 18 to 24 months. So there is an evident medical need, and as you can see also the non-Hodgkin's medication market is increasing annually, and 30% is accounted for diffuse large B-cell lymphoma. In order to improve the first-line therapy, a new cell line model was developed for diffuse large B-cell lymphoma, and different strategies to improve the first-line therapy was evaluated. The attention was towards HDAC inhibitors, which are known as sensitizers to chemotherapy. And valproate was the preferred choice, showing very good, strong results in the preclinical evaluation. And it is also known to have a positive effect on gene expression and cell cycle. It is a known drug since the 60s to treat epilepsy. And since it's a well-known drug, it has an established safety profile. And we were able to start a phase 1 study with the available drug available at the pharmacy. It should be noted that in the first line, the phase 1 study, we also established that valproate is synergistic with CD20 targeted therapy, which is in our case the R in R-CHOP. The phase 1/2a study showed a significant improvement of the overall survival, where we evaluated valproate in combination with immunochemotherapy R-CHOP. And as you can see, the 1-year overall survival was 100% and the 2-year overall survival was 96%. And in the study, we didn't have any control group, which is very common in oncology phase 1 studies. But we used the Swedish Lymphoma Registry for patients treated between year 2000 and 2015 and performed a matched pair analysis comparing patients with the same gender, age, and also disease status. And overall, we can show that we have a more than 10% increase in overall survival. And another statistic analysis showed that we have an 80% reduction of risk of mortality. And this is very strong results and should be compared to 30%, which is considered clinically 30% reduction of risk of mortality, which is considered clinically relevant. This data was used in a scientific advice meeting with EMA, which confirmed that Respiratorius may move directly to a phase 3 single pivotal study. With the addition of pharmacokinetic data for the new formulation we have been developing since. The new formulation is a combination of immediate and extended release granules and pellets, which releases the valproate over 6 hours, which means that it allows for twice daily dosing compared to the drugs we used in the phase 1/2a study, which were necessary to dose 3 times daily. The new formulation has a very distinct release profile, differentiating from all available valproate drugs on the market. And we are considering this an opportunity to actually place VL-001 as a new chemical entity pricing. And this has been confirmed with pricing experts. The PK study that we are planning to start as soon as possible, which will be performed in healthy subjects, and the required documentation has been filed to the ethics committee, and the submission of to the regulatory agency is due end of October, which allows for having all the approvals before end of this year. We have performed an analysis of the products with a similar strategy in terms of having an adjuvant to the standard first-line therapy with R-CHOP. And what really stands out here is that VL-001 outperforms the competition, both in terms of overall survival as well as lack of significant side effects. So this means that we have very strong data, and we have a strong confidence in VL-001. Over the years, we have accomplished quite a lot, quite a big deal, and we have a phase 3 study design, which has been confirmed with EMA. And we're also planning for scientific advice with FDA, because this is something that might allow for different regulatory strategies. So we are planning to do that. We have just recently accomplished the prototype selection, which is a very big step for Respiratorius, being able to have our own product. Before we only had very good results, but now we have also strong results and our own product. And we are planning to start the PK study as soon as possible. We have been able to have a patent granted over the overall concept, and we have a patent application on a new formulation, which is in prosecution in several countries, and we have made some progress over the last couple of months. Jumping directly to RCD-405, which is a novel and innovative first-in-class anti-inflammatory and bronchodilator for COPD and severe asthma, which is still in preclinical development, being prepared for a first-in-human study. Just to set the stage, COPD is one of the most common and rapidly growing diseases. And it is characterized by inflamed and constricted airways, and it is one of the leading causes of death globally. And current treatments are only to reduce risk of death, why this is there is a huge unmet medical need for a new drug. We have a lead candidate, which is RCD405, which is a new chemical entity, and we just recently had allowance for a patent. We have documented anti-inflammatory and bronchodilatory properties, and the physiochemical properties of the substance RCD405 is optimal for a dry powder inhalation formulation. Just recently, we announced that we have been able to accomplish strong preclinical pharmacology data in terms of an ex vivo study. You see some of the results from the study, where the study has been performed in different species, and what you see here is rat to the left and human to the right, human airway tissue, which has initially been constricted, and we can see the relaxation measured at different concentration. And this is very strong data, which will be supporting our first in-human study. Further milestones accomplished is that we have been in the preclinical data package is that we have performed initial safety studies with no critical findings. We have performed the ex vivo study with positive and strong results, and we have also in vitro evaluated the anti-inflammatory properties of RCD405. CMC and formulation, the production process is established, and we have a collaboration with Econovo on the formulation, and the initial fundamental finding is that RCD is optimal for formulation into a dry powder formulation. The value-enhancing milestones we can foresee of the next coming year, moving the asset forward to a phase 1 study, is that we have which will be the next step, is to initiate and complete the toxicology studies, which will be performed by inhalation in rat and dog. It's an expensive study, while we have been ticking the boxes prior to that, and it's a very important study. We will initiate additional clarifying mode of action studies, and we then tick the boxes in the coming toxicology studies. We will also initiate GMP production for the IMP clinical studies. We have been allowed a patent in Europe just recently, which is very positive, and the patent application prosecution is going forward also in other countries. And this concludes my presentation.
Thank you for an interesting presentation. You just completed a new formulation of VAL-001.
Yes.
How does this add to the competitive edge for the candidate?
It does. I mean, previously we have only had very strong results based on available drugs, but now we have a dedicated formulation which has a release profile which is perfect for this new use of valproate.
And you recently announced positive data on your candidate RCD405. How does this affect the project?
It is an important finding, and it's a reassuring that we are having a product that is effective, and next step will be to make sure it is safe as well. And this is the strategy is moving the asset forward in stepwise, and now we have the efficacy data, and we will then move forward to having the safety data also in the toxicology studies.
And the candidate is developed as a dry powder formulation together with Econovo.
Yes, it is. Yes.
What is the next step in the project?
It's also something that we move forward stepwise, And we have been able to have the fundamental data in forms of that it's an optimal substance for dry powder formulation. And as we progress in toxicology, we will initiate next step also in the formulation of the dry powder formulation for inhalation together with Iconovo.
Interesting, and thank you for coming here today, Johan.
Thank you.
Specifically, life science companies then usually need a lot more funds. As they mature into later stages, they start larger clinical trials and so on. Do you believe that the Nordic investor community, with the retail investors in particular, then can sustain the increased need for capital going forward?
It's always difficult to predict, but we have a strong Nordic capital market, perhaps the strongest market in Europe at the moment. And if you add to this that we have the biggest exchange within biotech in Europe, with more than 30% of all listings in Europe, and if you take the population here in the Nordic, we are not counting for 30%. So I'm really confident that we can support the journey for many of the life science companies with their future growth. Investors know where to look for biotech investments, but so do big pharma and big biotech. So it's not given that all the listings we see now and will come into the market and the exchange for the next half year will stay independent forever. There will be a lot of these companies that will be sold and taken off the exchange. So it is a mix, and we're looking forward to work with all the companies, either if they will make it big on the exchange here in the Nordic, or will be sold to some of our big clients like GenMap or Novozymes or Biogen in the US. And talking about US, we see more companies looking for the US to tap into more specialist investors as they grow bigger. So dual listing on NASDAQ in the US will be a part of many of the small biotech companies we have on the exchange right now. So if they want to make it big, some of them will also tap into our market in the US, and there's a lot of more investors there. So I'm confident that we can support the companies in the future.
And now I'm joined in our Lund studio by Peter Nässtedt, CEO of SenzaGen. Welcome, Peter, and please go ahead with the presentation.
Thank you very much, and welcome from my side as well to my presentation about SenzaGen. So, my name is Peter Nässtedt. I joined as CEO of SenzaGen 1.5 months ago. I have a broad commercial and leadership experience in the life science sector. I've worked with both organic growth projects, establishing sales in new territories, working with channels and so on, but also with acquisition-driven growth, where I have worked on several large and small and acquisition projects, including both finding targets and performing them and integrating them. I have served as CEO of Probi, also a publicly traded company. Between 2014 and 2018, we had growth from 100 million Swedish kronor up to 600 million. And previously I worked in larger groups like GE Healthcare Life Sciences and also in Trelleborg Marine Systems. I worked here in Sweden. I've always worked in international positions, I should say. I also spent 4 years in US. I joined SenzaGen already in 2018, but as a board director. And I've worked in operational roles in SenzaGen, primarily with the commercial development and strategic development. My educational background, I have a master's in chemical engineering and a degree in business administration. So, what is SenzaGen about then? So SenzaGen has a vision to replace animal testing with leading in vitro technology, thereby establishing new industry standards and becoming a leader in the industry. And we will do that with leading technology. So we have already deployed leading technology by developing the GARD platform. The GARD platform combines cell biology, genomics, and machine learning. And we have 2 different tests, one for measuring skin allergens or identifying skin allergens, and one for identifying respiratory allergens. These tests have very high performance and very high human relevance and high reproducibility and applicability, meaning that they can be used for many different types of substances. We have complemented our strategy and also have other in vitro toxicology testing services like skin irritation and corrosion. We have started our commercial development with SenzaGen. We are focusing on 3 main markets, so Europe, North America, and parts of Asia. We deploy a direct sales strategy where we're working directly with a number of customers, but we have also engaged a number of distributors, and I'll come back to our sales model in a second. What is new is that we also have an active acquisition strategy that we're implementing now. And the market that we're operating on is growing, and it's growing because of several factors. One is regulatory. It is, we have animal testing ban in Europe for cosmetics, for instance. We have similar bans in place in a number of states in US. There is an ethical concern and an active social responsibility work in corporate world, which is also aiming to reduce animal trials. And there is, of course, a performance-related component of this. The world needs better tests with better human predictability. So I'll go into our growth strategy, because we have worked a lot with our strategy to devise something which will make us grow even faster. And we already had an organic growth strategy. Our organic growth is about establishing the GARD platform with direct and distributor sales, and also a select number of CROs, which I will get back to in a second. We complemented the organic growth with acquisitions and also by deploying new tests ourselves. And on the acquisition growth, we're looking for companies in our industry which can complement SenzaGen, either in terms of geography and reach, so in other words, reaching customers in areas where we have a hard time reaching in ourselves, or making our test portfolio broader. So having a broader portfolio that will enable us to target more customers and also satisfy their needs for other testing purposes. To develop our thoughts a little bit around our growth strategy, first of all, one important component of our organic growth is to achieve a OECD test guideline status of our platform GuardSkin. And this validation exercise that we started a few years ago has now resulted in a very good opinion from the independent expert group of EU called ESAC or ESAC. And this opinion says that GuardSkin meets the standards in terms of human predictability and also meets the standards of reproducibility. And they give a very firm recommendation that GuardSkin should now become a OECD test guideline test. So this is a big achievement for the company. This news came in this summer. Once adopted as an OECD test guideline, it will expand our market for testing because it will be easier for our customers to use results from the Guard platform in their regulatory processes. So, as you can see from this illustrative picture in the bottom, the market that we've been operating on up until now is for non-regulatory sensitization testing. And we are now expanding that, or have expanded that, by offering 2 complementary skin tox tests, one for irritation and one for corrosion. And with the OECD test guideline, we can reach a market of around $550 million, and thereby significantly expanding from our current or earlier position. To reach a larger part of the toxicology, in vitro toxicology market, we see that we can offer ourselves a number of OECD validated tests. So that's what we are doing next, and we have a number of new tests in the pipeline. Then the potential market of 6 billion is quite attractive, and I'll describe it in more detail in a second. And to address the full market, we do need a number of complementary tests, and that's where we can see that acquisition strategy will play a major role for us to address that part. So, as I said earlier, I would want to explain a little more about our business model and sales strategy. So first of all, for the Guard tests that we have developed ourselves, which are high performing and with broad applicability and reproducible, we have a direct sales approach. So we have developed and deployed our own sales force. They are working directly with some of the leading companies in the world in implementing the Guard test. We have in certain markets chosen to deploy distributors. We've done that in a number of European markets, but we've also done it in North America. Then we are also working with a select number of licensing partners, or CROs. These companies complement the SenzaGen lab capacity for Guard testing. And we have signed agreements with leading CRO companies, I should say. So that is our strategy for the Guard test. Now, when we have started to work directly with customers with Guard, we saw that they have a number of other testing needs. Associated with identifying skin allergens, you also need to do other toxicity testing. That's why and then the other learning we had is that we have very high standards in our lab. We have one of 2 GLP certified labs in the Nordic region. So we have an opportunity to fulfill customer needs with broader testing possibilities. That's what we've done now. We are offering 2 new tests, one for irritation and one for corrosion. And going forward, we will expand our test offering, and I'll get back to in a little bit what type of test offering that can be. Finally, we are working with acquisitions. So we have learned how this industry looks. We have seen how the landscape is establishing, and we see an opportunity for complementing SenzaGen with a number of acquisitions aimed at broadening the test portfolio and also improving our geographical scope. What is important with our acquisitions is that we have a requirement that the customers or the targets need to have reached profitability, and they need to be growing. So this strategy that we devised, it's possible to implement it thanks to a strong market position that we already established with GARD. So why do we have a strong position with GARD? Well, first of all, we have a portfolio now. So first of all, we have a GARD skin test. It is positioned as a very accurate, reproducible, and broad applicable test. It's used in cosmetics, chemicals, and pharmaceuticals. We've made an adaptation of GARD skin for medical devices so that it can be used with solvents and detect leachables from devices. We also have 2 risk assessment tools based on the GARD platform, GARD potency and GARD dose response. GARD potency is a qualitative determination of a weak or strong sensitizer. When we worked with customers, we did see a need for clients. They are discovering that, oh, I have an allergen. Then they would like to find out, well, what is the situation with this substance? What is the point of departure when this becomes a problem for me? Then you can measure different concentrations. That's what we are doing with GARD skin dose response, and we have high interest from several industries, including pharmaceutical, medical device, cosmetics, and chemical industry for this test. It is also the only quantitative in vitro test available. Finally, we have the GARD air test. It is a unique way of identifying air sensitizers with an in vitro test. It has a very high specificity, so we have positioned the test as an opt-out tool during product development. In other words, if you identify an airway sensitizer with GARD air, you should not continue to develop that product. So a little bit of history is that the in vivo tests are not very human relevant. They reach around 70 to 75% human relevance in terms of accuracy. There are a number of ethical and regulatory issues with animal tests, and they are very time consuming, both in terms of the test itself, but also in applying for ethical committee and these type of permits to perform the test. We have a first generation in vitro test. They often build on one single biomarker. You can do a qualitative potency or risk assessment, and they are often combined in approaches. The GARD platform reaches 90 to 95% accuracy because we have 200 genes in our signature. So we are measuring a very broad spectrum in the allergen space. That's why also customers are interested in the GARD platform, because of the accuracy across the applicability domain, so for many different types of substances. It's also possible to make a quantitative risk assessment, as I alluded to earlier with the test. And as you can see, the broad applicability and accuracy also plays a major role in that GARD can assess difficult to test substances or challenging substances. For instance, metal salts, compounds of variable and unknown mixture, complex mixtures, surfactants. These are substances which are difficult to address in other tests, but we can do it in GARD. So that's the GARD platform. That is where our starting position. It has built us a good foundation and a growing customer portfolio. In addition to GARD, we're going to expand with our CRO test portfolio. So currently, we've already launched the irritation and corrosion test, and we also launched an irritation test for medical devices. Going forward, we're going to launch a complete medical device biocompatibility testing kit. We want to do that before the end of this year. In addition, there is a number of sort of related test items in terms of using cell-based testing in phototoxicity and cytotoxicity. These are also endpoints that clients test for when they are testing for allergens. So it's very natural that we offer these tests, so that is in our pipeline. The other tests, there are very exciting developments going on in, for instance, genotoxicity or organ toxicity. These are areas where there is new technology deployed. We think it's very interesting, and we can see that that's a path where we will direct our acquisition activities. So we've started our commercial journey. We have every half year we have more new customers than we had the half year before. Plus, we have an increased customer retention, so we have more and more customers returning. This is creating what I call a snowball effect, and now we want to see that snowball getting larger and larger. So we also see a substantial increase in customer inquiries recently. We have, based on the feedback, expanded our test offering, as I told you about earlier, and then we have widened our distribution capacity with an agreement with Enthalpy in US. We have expanded our agreement with Charles River, one of the largest CROs in the world. We did a very high-profile project with American Research Institute for Fragrance Materials, or RIFM, where we have tested fragrance materials on guard. And RIFM has 70 membership companies, so this is an excellent opportunity for us to present our capabilities in the space to them. Just to get a little bit of a feel for what type of customers we have, we're working with medium to large size leading companies in the cosmetics, medical device, chemical and pharmaceutical space. And we've had scientific presentations with Essity, a Swedish health tech and consumer product company with 114 billion Swedish kronor of sales. We worked with Sonova, which is a Swiss medical device company with 2.6 billion Swiss franc in revenues. And we worked with Corteva and also did scientific presentations with Corteva. Corteva is one of the absolute leading agrochemical companies in the world, previously also known as DuPont. So these are the types of customers that we're working with. We're very pleased with seeing a good growth of our and expanding customer base. I want to go into the toxicology testing market, and this is, as I said, a very interesting market. It is growing with between 5 and 10%. We're estimating value of, it's estimated that it will be worth nearly $9 billion 2023. And we are already operating in the fastest growing segment, which is skin sensitization, corrosion and irritation testing. It is estimated to be worth $550 million by 2023, and it's growing by 9.5% per year. As I mentioned earlier, we see an opportunity to expand our offering in cytotoxicity and phototoxicity because they are cell-based assays, and they are also related to testing in the skin allergen space. So this is what we're targeting with our organic growth strategy. And the other sectors of this industry, which are also very interesting, is what we want to address with acquisition growth. And all sectors, chemical, pharmaceutical, medical device and cosmetics, have similar needs around toxicity testing, which makes this a very interesting market. The market growth is driven by several factors which are all sort of working in the same direction, and that is to reduce and replace animal testing. First, it's the regulatory environment, where we have the cosmetics ban in Europe. We also had a resolution by the EU Parliament 3 weeks ago, where they issued a recommendation or resolution to the member states to accelerate the switch from animal testing to in vitro testing. Ethical considerations, we have the corporate world is taking a larger social responsibility and have goals set in reducing their animal testing. And then we have scientific, so the tests that are on the market are not always good enough, so there is a need for more accurate tests, more human relevance in the tests. And then there is a need for more economic testing as well, so tests that are quicker to perform and tests that are less costly to implement. That was my last slide, so thank you very much for listening to me, and I welcome any questions from you.
Yes, well, they're coming right up. Like you mentioned here, you took over as CEO in August. If you were to describe SenzaGen's potential, how would you do that?
Well, I think SenzaGen, with the market-leading technology that we have an opportunity to become a leader in the in vitro toxicology space for animal-free testing, and that's our goal to do with both organic growth and acquisitions.
And you talked about the new strategy, but just to summarize it a little bit, how does that differ from the strategy you presented in 2019?
So we've developed a strategy and made a higher emphasis on growth, or even higher emphasis on growth. So we had a great organic growth strategy, but we have built that out with offering a number of additional tests. And now we've also added acquisition growth. So it's a more growth-oriented strategy.
And finally then, in the short term, what are the most important milestones for SenzaGen?
Yeah, we're working on several fronts, but short term I would like us to grow a little bit faster than we've done before, even faster. I'd like us to implement a number of these new tests that we've had in the pipeline. We should be working and seeing this OECD test guideline status in the near term. It's a very near-term priority for us. And finally, we have an active acquisition pipeline, so I hope that we can bring something to close also in the near term.
Well, we look forward to hearing about that.
Okay.
Thank you so much for coming.
Thank you very much.
The next presenting company is Cereno Scientific with CEO Sten Sørensen. Welcome, Sten.
Thank you. So I'm very pleased to be back and to present Cereno Scientific's exciting development over the last period. So what are we doing? Cereno develops novel drugs to transform treatments for common and rare diseases. And cardiovascular disease is the #1 cause of death globally, killing nearly twice as many people as cancer. There is a significant market need due to the high mortality and very high morbidity adversely affecting patients' quality of life. We have a pipeline portfolio, a phase 2 program with our lead product, CS1, in rare disease, pulmonary arterial hypertension. And we have 2 preclinical programs in cardiovascular disease. Our global footprint is, we have our head office in AstraZeneca, as biotech hub in Gothenburg, Sweden. And we also have a US subsidiary in Boston. In addition, we have R&D collaboration with University of Michigan in Ann Arbor. And as you will see, we also have our phase 2 program to be initiated in the US. Cardiovascular disease is a group of disorders affecting the heart and the blood vessels. By 2030, it's estimated that around 22 million people will die of this disease. And as you can see to the right, it has tremendous cost to societies, here illustrated by European Union's and US cost. These are estimated to double by the mid-2030s. Looking at Cereno's journey since the start, from discovery, we have now developed the company over the last 2 years, progressing our research and broadening our pipeline. So today we have 3 programs in the portfolio. We have a well-defined target market for our lead candidate. The valuation of the company as a result has increased, and our ongoing activities are fully financed. Looking at the portfolio, we target with our CS1 lead project, which is an epigenetic HDAC inhibitor, the rare disease pulmonary arterial hypertension. And we're about to initiate that study after FDA approval recently. The 2 preclinical programs is a prostacyclin analog, CS585. We have agreed with University of Michigan to evaluate this program over 2 years, and we have the right to in-license it exclusively for further development and marketing. The other program is CS014, and it's also an epigenetic HDAC inhibitor program as CS1 is. And both these preclinical programs are targeting cardiovascular disease. Looking at the lead project, CS1 in development for rare disease, pulmonary arterial hypertension. This is a very severe disease. It's an orphan disease affecting less than 200,000 people in the US. And it has a prognosis similar to many cancers, very poor prognosis. And eventually it leads to right heart failure and death. And the real cure is lung transplantation, but very few patients reach that stage. So, and the drugs don't really do more than lower the pressure in the pulmonary arteries. So there is a need for new disease-modifying drugs, and we're trying to do that, and we have a very innovative approach. Looking at CS1, we aim to offer a better disease-modifying treatment for this rare disease. And the characteristics which have been documented over extensive studies in preclinical work, mainly animal studies, have identified 4 characteristics, efficacy characteristics: anti-fibrotic, anti-inflammatory, pulmonary pressure reduction, and anti-thrombotic efficacies. And this fits like a glove with the pathophysiology and pathogenesis of pulmonary arterial hypertension. And this development could fit the need for what's needed in new advances in therapy for these patients. So our lead compound, CS1, has demonstrated positive results and solid results in preclinical and animal studies. And we also have done a phase 1 trial with good safety and tolerability. We reached our targeted pharmacokinetics, and very excitingly, we lowered the clinical important biomarker PI1, which is important for antithrombotic and fibrotic effects. Now, we recently got approved our phase 2 program by FDA, and it will be run in the US in 6 centers, and will have a principal investigator in Raymond Benza. When this study is done, hopefully positively, we'll move into phase 2/3 program. So FDA approved our IND to go ahead and study CS1 in these patients. And here's some more information. So we will look at standard efficacy endpoints for this patient group. We'll also look at the validated risk score, pharmacokinetics, and we'll look for the best dose to move forward into the next study. It will include 30 patients at 6 different sites, and top-line results is expected by the end of 2022. Raymond Benza is a leader in this field, and he will be the principal investigator. Some more details about the study that we haven't presented before. So the patients that will be included in this study are patients that are on mono or double therapy stable, but who are symptomatic and have a risk for clinical worsening. It will be adult male and female subjects, quite wide age and BMI range. And the patients after a screening period will go through right heart catheterization to implant a pressure device, cutting-edge technology, CardioMEMS. And then a baseline period of 6 weeks will follow, where the patient will familiarize itself with the technology, and also we will have baseline pulmonary arterial pressure measurements. After this period, the patient will be randomized to one of 3 dosages: a low dose, intermediate dose, and a high dose, based on our animal studies. The treatment period will run for 12 weeks, and then there will be a follow-up period for 2 weeks. And during these 12 weeks, we'll measure the pressure, of course, but also take laboratory data for other matters. So we have signed a collaboration deal with a partner here, the Abbott, a large pharmaceutical company, with using their cutting-edge technology, CardioMEMS, which is an implantable device that you put into the pulmonary artery. And that with that device, you can make readouts when you want during the 24 hours, and you can transfer remotely the captured data to the physician's office, and we can get very good data and readout. And this way to study use this technology will make the study cost efficient and also improve our ability to get good data. Let's move on to our preclinical program. So we have, as I mentioned, agreed with University of Michigan to have an exclusive program to evaluate stable, selective and potent prostacyclin agonist, CS585. And it has potential to significantly advance treatments within cardiovascular disease. The other program is a compound that we acquired from Emeriti Bio at AstraZeneca Hub. So we have two preclinical programs, and we signed an agreement with University of Michigan to evaluate these two compounds over 24 months. And this agreement was put in place during the spring. So 24 months from May, we'll be able to have packages, if everything goes well, to move these compounds into man in the first clinical trial. The program is led by Dr. Michael Hollenstadt, active at University of Michigan, and he's also Director of Translational Research for Cereno. Now, let's look at some achievements that has propelled the development of Cereno over the last year. So we have strengthened our team for clinical study in preclinical phase programs. We have expanded patent protection for CS1 and CS585. So CS585 got US patent this year. CS1 had US patent before, but got Australia, Japan and Russia patents. And also recently the whole European Union, 15 countries in Europe. We signed, as I mentioned, an option agreement for in-licensing of 585 and initiated preclinical programs for CS585 and CS014 at University of Michigan. We entered the collaboration agreement with Abbott to collaborate in the phase 2 study with CS1 and use their cutting technology CardioMEMS pressure technique. We obtained our IND acceptance for the phase 2 trial that will be run in the US for our lead project CS1 and the phase 2 study in PAH. And then very recently, actually last Friday, we had a successful share issue with warrants of series of TO1 that were exercised to nearly 97% and brought in 95 million Swedish kronor to the company. So these activities and this last financing round with the warrants has now fully financed our future development of the company. And you can see the development here of market cap and share price over the period. I should say also that there is a second warrant package a year from now in September next year that is potentially bringing in another 114 million. So our focus now moving forward is the CS1 phase 2 study, get the study sites up and running, recruit the first patient. We will communicate and position Cereno in the cardiovascular space scientifically, and we will push our preclinical programs forward. In addition, we'll work on our IP protection for the company. So this is an overview over our focus over the last 2 years and the 2 years coming forward. And if everything goes well here, we are in clinic with the 2 preclinical programs by 2023, and we'll also initiate our next phase 2 program with the lead compound. That ends my presentation. Thank you.
Thank you, Sten. Seems as if you've been really busy during the summer.
Absolutely.
And now you're even having an even busier autumn.
Yeah, you know, it's when you drive many programs, the positive thing is that you know, many things that can grow in value and so on, but also results in a lot of activities. And now, as I mentioned, our focus is to get ethical approval and contract the sites and recruit the first patients for the lead program and push our preclinical programs forward.
And you also mentioned the warrant issue of TO1, or the exercise of the warrant.
Yeah, I'm very happy about the very strong support that we are getting from the shareholders that came in a year ago, and also the existing ones. So feel very positive about that, because now we can focus on executing our programs.
And you also mentioned the exercise of the warrant TO1, which provided almost or a little more than 95 million kroner. What does that mean for Cereno?
Yeah, well, it gives us a very good feeling that a very strong support from our existing shareholders, those who came in at the last share issue a year ago, have continued to support the company, as well as those who were there from the beginning. And now we can focus on executing our programs as we are fully financed for what we have planned.
Yes, and final question. You said that you're aiming to present top-line data for CS1 in H2 2022. What will happen after that?
Well, yeah, so that's the real task here, is to actually be able to pursue the clinical trial on PH with CS1. And we hope to be able to do that on time. And, we haven't moved our top line target date, which is, as you say, during H2 22. After that, depending on if the data is good, which we hope, and actually already now we're going to initiate planning for the next trial. But we need the data from this trial to be able to finalize that planning. But that planning will start as soon as we are up and running with this one. So next year will be a lot of work on the next trial, as well as executing this one.
Sounds really interesting. Thank you so much, Tim.
Thank you.
next presenting company will be Aptahem, and with us we have CEO Mikael Lindstam. Welcome, Mikael.
Thank you so much. Nice to be here. Well, I'm the CEO of Aptahem, and I'm here today to tell you a little about Aptahem. But for you newcomers, I will give you a brief about Aptahem, and then a little where we are and of the latest findings, and also round off with a bit of the future. So hang on. So Aptahem, the mission of Aptahem is to develop aptamer-based drugs, especially for the treatment of life-threatening diseases. Apta1 is our lead candidate, and it's being developed towards acute application, and in this case to prevent the damage of organs and tissues in sepsis patients. This is a very big job and very interesting to do. Aptamers, which I just mentioned, is a fantastic group of different drug candidates that we can manufacture, and aptamers are so unique that when you work with them, you can build them from DNA or RNA fragments. And these fragments can be looked upon like Lego pieces or actually pieces of a puzzle. So you can match and mix them into a unique target and work on that. And Apta1 is an RNA aptamer. And the uniqueness, I just went into that, is that we could manufacture interesting aptamers or drug aptamers for targets that you have not been able to really reach or approach so far. Meaning we could actually approach totally new treatments for sicknesses that doesn't have a solution today. So that's a fantastic ability with aptamers. If we then go back to our lead candidate, Apta1, we see here that we have, including with its brothers and sisters, a multifunctional portfolio. And especially that we can apply it to several different inflammatory diseases. And we're working towards sepsis, which is a syndrome sickness. What else about Apta1? Well, it has brothers and sisters that I said. And if you look down there, you see we have Apta1, Apta2, and Apta3, sisters and brothers. We don't know much about them today in the case of what we're doing here, but we have touched a little with Apta2 in the inflammatory department. And we see that it looks very much like Apta1. So the future holds interesting pipelines for us, and also that we could open up when we have the economy and the people to do this. But we're looking forward to do things like that. And lately we have further pinpointed our novel mechanism for Apta1. And now we have so much that we can go down to molecular level and see that Apta1 hinders thrombin from activating PAR receptors. And this is a fantastic finding. And it activates these PAR receptors on a very, very narrow part. And I will come back to that in a few slides. And by this hindrance, we can see that we can cause a rapid anti-inflammatory and anticoagulant or antithrombotic effect, more correctly. And this is the key to how good Apta1 is working in the sepsis conditions, or sepsis-like conditions that we have tested so far. If we go on to next here, Apta1 again, I will not go into much, it's a take-home message here. And because we can bind to so much specific targets, or subtle targets as I mentioned here, results in a lot of different advantages. And if we look at the two most important things here that I just mentioned, the novel mechanism that we have for this thrombin inhibitor is that it doesn't resemble the typical thrombin inhibitor that you have in the clinic. And this is the uniqueness of Apta1. And the specific one is that it doesn't cause bleeding effect. Fantastic. And there are some other, of course, advantages too, but I'll come back to them in the second point here. When it interacts with the coagulation factor in the thrombin, then we see also that the hemostatic functions remain unaffected. And that is the key here with Apta1. It lets the coagulation system just move on, but it's pinpoint one part that actually is the villain behind triggering the inflammatory and the hemostatic mechanism in the body. Fantastic. I've been talking about anti-inflammatory and anticoagulant, and we have some more effects. If we look at the picture to the right, this is how we define Apta1 today. It has two legs. One leg is the modulation of thrombin, I've been talking about that, and we have the two first ones of blood coagulation and hyperinflammation to inhibit this. We also have a very important part, it's called promote the reparation of tissue, and this is for sepsis extremely important, because when you're struck by sepsis you can have an hyperinflammatory response and the body reacts very strangely, so much that it starts to leak from the vascular system, so blood leaks out from the vessels. That means you lose blood pressure, you lose oxygen, and in the end, it can be so bad that the organ dies due to oxygen deficiency. And this is other and third very important part about Aptahem. If you look at the right leg, you see that we also induces a stimulation of pentaraxin 3. I have always tough to say this word. Anyway, we stimulate this more than 10 times than the body usually can do. And this is the second secret sauce recipe. And it also stimulates the same parts that I've talked about, the modulation of thrombin, but with an added fact. It identifies and eliminates pathogens. And pathogens are virus, fungi, bacteria, which is the most common in sepsis. We are still working on looking how to define this and find more evidence for this part, but so far it's very interesting what we have seen together with collaborators. Anyway, if we just take a look at the whole picture here, Aptahem modulates inborn immune response. What does it mean, inborn? Well, it's the body's own immune system that are under supervision of Aptahem. Aptahem is a management consultant of big range. It goes in, tells what to do, supports, stimulates, modulates, and something happens. So it doesn't add anything more than just good advice for the body to regain the balance of the body system, the immune system. A fantastic feature. And what better to use it into a sepsis syndrome? Absolutely not. Much good it is. Anyway, so I'm talking about the novel mechanism and the pinpointing with it. This is a little visualization of what we are doing right now, or what we have seen together with a collaboration with Örebro University. We have Aptahem coming from below up to the heparin binding site. This is, the thrombin is a coagulation factor, and it contains a lot of different sites. And one of those sites are called the heparin binding site. And Aptahem goes into a part of this heparin binding site. Just a little one. I've been very narrow, I told you before. And this is enough to stop the activation of PAR receptors. And here's the secret sauce recipe again. This is what happens, is that we stop the body system to tickle inflammatory responses and coagulation or hemostasis. And what happens when we don't? Well, we have, and in this case with Aptahem, a very rapid anti-inflammatory and anti-thrombotic effect. It's a fantastic thing. And that's been done in collaboration with Örebro, which have been stellar in this way. And we're still doing more. This is one mechanism. There are more, and we're looking into those as more. But this is a very important one, which explains Aptahem's fantastic effect. And effect we have, and why are we doing it? Sepsis, I talked about this, and I talked about the vessels opening, and you lose oxygen in your organs, meaning your organs are in threat to die, actually. And if you don't die and you survive, you can still have an aftermath of brain damage. In case of cognitive problems, you need to maybe replace your kidneys or something like that. So it's nothing to play with. So the faster you put in a remedy or a therapeutic to take care of a septic condition, the better for the patient, and of course for survival in the end. And of course, as you see, there's a lot of people on the right side that are struck by sepsis every year, and the cost associated is enormous. And this is not the full cost, of course, because the socioeconomic costs are even bigger sometimes, and the personal suffering. We should remember that. But there is a market potential. And with that, well, it's not only us doing this. There are others working on solutions to fight against sepsis. And if you look here, that's maybe not too many out there, but still, we are not alone. That means there is a need to do this, and others have realized that as well as we. And we're now gaining towards phase 1, so I hope to put a 2 on phase 1 there when we come into after the summer next year, if we keep the schedule. And just to say something else about Aptahem, and I feel is that recently, last year, they approved an Aptahem drug in Europe. And that is very important, because as soon the regulators have taken them to them, and the national offices dealing with regulatory, then they open up doors for an understanding and a positiveness towards this type of drugs. It's very novel drug chemistry or area where Aptahem are in, so we should remember that. But the more is done, the easier it will be, at least to convince the fantastic properties of Aptahem. And just to mention something here before I go on, is that what we see with Aptahem is that it offers a very wide, if not totally, covering therapeutic effect on the sepsis syndrome. And that is something that we see from all our studies that we have done. But of course, there are may others out there that say that, okay, we do that too, but so far we haven't seen that. But that keeps us moving on and working hard with Aptahem. And we talk about market potential. We talked about people working on this to find solutions. And here's an evidence for the potential market. It's multi-billion US dollars at stake. And of course, if you look at this part here, down here, market share of patients treated with immunomodulatory drugs, that is where we are. And if you look at the figures for Europe, 1/3, US 1/10, and 40% in Japan, that means there's an even bigger market potential ahead of us, because the focus right now is in that part. And here is what Aptahem also is a position, so to say. And so with all this potential market and so forth, we need partners, because going to market costs billions of US dollars. And there we are. So we can't do it ourselves, that's for sure. We wish we could, but there's money and expertise that need to come in from a partnering. And if we look at this, we need to protect our product. If we don't protect it, no one will take it, because who will invest billions of dollars and then someone else do it and take the market? So this is a very important thing, to have patent protection. And we have two patent families. One is on the free structures, and those are fully granted. And then we have patent family 2 for Aptahem, that is a therapeutic use patent. And there we already have a granted patent in the United States and in Europe, and we are validating Europe now. And we are also looking, as you see here on the map, several other heavy states for market and developing Aptahem further. So if we go on to the collaborations, well, because we are not here today by our own speed, others have been part of our success, and here we have 3 now that we are working with currently, in Örebro in the middle, I've already mentioned. They've been stellar in looking at this particular modulation and mechanism that we have right now. Seattle Children's Research working with real sepsis models and also pathogens. University Health Network in Canada are working on lung models, virus-induced lung models. That means corona, SARS, and others. So we're looking at other pathogens and to see how that goes. And we are having quite nice results from there, but we are still waiting for final reports to really claim what we are saying here. And Örebro, I mentioned before, very good and very important for us, all of these in our work for bringing Aptahem, not even scientifically, but also into the regulatory and clinic. It's very important for us to have this. And here's the team at Aptahem, some of them, or its management team. Most of you have seen them before, and I will not say so much about them, but you can look more on the homepage. But we are a small team, semi-virtual company, and we're working a lot with third-party providers, very good ones. So there we are. And just to round off, I promised a little outlook, and here we are. We are ready to go into clinic 2022 after the summer, as it looks now, and what we communicated so far, we are staying on plan. And if you look at the outline timeline there, we see that we are still having some work to do. It's regulatory, it's manufacturing GMP material for the company, and to go into clinic. And phase 1, you see, it's a longer stretch, because we're also planning a challenged study in healthy individuals to see an early response, how Aptahem works under a stimulated inflammatory response. And if you look at the deal and partnerships wide stretch there is, because we are constantly working with this. And there are, of course, inflection points now, as after phase 1, and specifically after a phase 2, which is a proof of concept in man. And I also shadowed a little the part of phase 2. We are not there yet, and it's very preliminary, but I just want to show you where we are heading. And during this time, we also would like to find a partner. We're working hard on that. But phase 1 ready, you see the timeline, we have communicated, and we are moving towards there. Anyway, we are starting to get really ready for phase 1. We're working hard on partnering, and we are also working on the scientific presence. And I just mentioned it a few minutes ago that this is very important for us to support the regulatory, clinic, but also show that we are a force to count on when it comes to the science we are developing here and the product. It's very important to have that. And there's no better than to have a publication out there, and we're working hard with our collaborators to put them out there now. It's massive amount of results we have take a long time to assemble, but we are doing that, and it's a validation, as I said, out there that we are seeking from that. But we have fantastic result. But that I will stop talking and say thank you so much for your attention, and see you soon, and bye.
Thank you for your presentation.
Thanks.
And recently you had a development breakthrough in the collaboration with Örebro University. What are the possible regulatory and partnering implications of this?
Well, I mean, it's about the definition. I mean, we have defined even more down, and as we have communicated at a molecular level of where we are today. And I mean, we see that it affects the whole system, but without affecting the cell. So affecting just a key important mechanism that is responsible for triggering inflammatory and hemostatic reactions. So we actually hinders that, and we can actually turn around and have an anti-thrombotic and an anti-inflammatory response. And that's fantastic. And recently today, I come from a meeting here a few hours ago, and we was shown even more results, new results that even more strengthen what we have said here today. Fantastic results. So we are now getting so much results, so we are splitting perhaps up them in more potential scientific publications. It's such a massive amount of results, and they are all pointing in the same direction, which is fantastic. So it's a really, really nice collaboration with Örebro. And regulatory, of course, to define the mechanism for a partnering possibility. Of course, you're always asked this, and that's very important to have with you. So now we have it. We knew it from all the studies we've done, but we couldn't put the finger on where it was until now.
And you're now approaching your biggest milestone, clinical studies with Aptahem. What medical void do you intend to fill with this candidate?
Well, yeah, it's a slam dunk kind of. Yeah, the void is that sepsis is the biggest killer in the world. We know this, and it's also called the silent killer because you can come into the hospital and be dead half an hour later. And a lot of other sicknesses are triggering sepsis, like pneumonia, and we have seen COVID, of course, and so forth. So it's a huge void, and it has had no real treatment options since they began focusing on this area, because it's such a multi-processed functional syndrome. We don't say sickness because there's a lot of different things happen. So that's what we're targeting here.
And what are the most important steps for Aptahem in the short and medium term going forward?
Well, going forward, yeah, I just presented here that we are looking at putting together the full bag of needed information and data and so forth for the regulatories to be able to go into first-in-man studies here. And we're putting a lot of focus there. But in general, we kind of close the preclinic for the regulatory to go into to clinic here, but we're still doing work, but it's to add and support. And the talks we are waiting for now will be the final of the preclinic, and then we are looking at the regulatory part in a way of how you formulate and how you argue for your product, of course, very important. So that's what we're doing now. And I'm also negotiating with providers for performing the clinic study at the moment. So it's full speed. And we need to have this clear, and I have some really good candidates. Looking forward to present them at a time point when we are done with that.
Yeah, and we look forward to follow this, and thank you for coming here.
Thank you so much. My pleasure.
Next up from our studio here in Stockholm, we have Annexin Pharmaceuticals, and presenting is CEO Anders Hägerstrand. Welcome, Anders.
Thank you very much. So, I'm going to talk to you about Annexin Pharmaceuticals. I will start with presenting a little bit of an overview, then I'll talk about the rights issue that we just announced. I will talk about who we are and what we do, what are basic science and our key focus in the clinical development, and where we've come so far. The overview is that we are listed on First North. We are Stockholm-based. We have a semi-virtual organization, which means that we are few employees, only 5, but we are world leading in the field of Annexin A5. I will come back to that. Our main product is NXV, which is, as you can see in the little vial down there to the right. It's really going to be a first-in-class biological product. We have produced it under GMP conditions, which means that it's fully fledged for clinical purposes, and we have to do some modifications on the process. It's only minor so to take it into phase 3 into the market. We have strong IP protection, protecting the product as such and the production of it and the use of it in many clinical indications. We're happy to have announced fairly recently that we have finished our phase 1 trial, which opens a lot of doors for us. We have a focus on ophthalmology, an indication called retinal vein occlusion, which is really our key focus at the moment. We are starting an imaging trial in the Netherlands. I'll come back to that. We hope to start a phase 2 proof of concept trial with the financing that we're currently looking at. So the rights issue is the price is set for 125. With that issue, we will raise 73.5 million. It comes with a warrant issue in April, so shareholders will have the opportunity to invest even more in the company, which would give us another 18 to 29 million, depending on the stock price at the time, to which the owners will get 70% price reduction on the stock price. Our largest shareholders have committed to exercise all the warrants acquired in that rights issue, and the whole rights issue is actually guaranteed as a whole. I think most importantly, probably to the right, is the subscription period starts at October 20. We'll present the prospectus in October 19th, and the end of subscription is at November 3rd. The proceeds will be used first and foremost for our phase 2 proof of concept trial in RVO. Then we'll obviously work with the product further, with the business development and protect the patents and so forth, amounting to the total 73.5 million. Well, the team here is really good one. Myself, I'm the CEO, background MD, PhD from Karolinska, worked in Astra, and that became AstraZeneca. Then I joined this sort of the life science community in Sweden with startup companies. Have sort of led 2 of them to acquisitions. I joined the board of the company in 2018, became the CEO in 2019. Anna Frostergård is the CMO. She's working in this field of Annexin biology for I think 15 years or so. Susan Sachdev is our CEO with a background from Pfizer and CROs running clinical trials. Henrik Palme is our CFO with a lot of experience from different life science companies in Sweden. Ulrika Handal is our key individual who knows everything about producing biological material for the clinic. And our therapy head of ophthalmology is not yet announced, but he's a very senior ophthalmologist with a clinical and pharmaceutical company experience. I'm not going into details about the board, but we have a representative and the chairman, Haxel, who's done a tremendous work in the US, first in AstraZeneca or Astra, and then in the US, taking company from private to public into NASDAQ, and with a medicine registered for treatment of Parkinson's. But we also have representatives working with pharmaceutical development. We have investor relations. Carl Fredrik is representing the owners. Johan Frostergård is the founder, and Morten is the key business development expertise in the company. Now this sort of switch gears a little bit and I'll show you a movie that describes our key focus.
Over 16 million people worldwide are diagnosed with retinal vein occlusion. This eye disease, also known as RVO, is one of the most common causes of vision loss. At the moment, no treatment is capable of helping these patients before complications occur. To solve this, Annexin Pharmaceuticals is developing the biological drug candidate ANXV, a recombinant human protein Annexin A5. RVO can be caused by sticky red blood cells, cells that are defined by the lipid molecule found on their surface, PS. These sticky cells can form a blockage in the vein at the back of the eye, resulting in inflammation, cell damage, swelling, and pressure on the nerves. Such a blockage can eventually lead to loss of vision and, in some cases, blindness. This is how ANXV is expected to work. After being introduced into the body, it finds the blockage in the eye vein. It then proceeds to build a shield, covering the PS on the sticky red blood cells. This ANXV shield stops the cells from sticking, thereby decreasing the blockage, while also helping to battle inflammation and repair damaged cells. As blood flow improves, the swelling and pressure on the nerves starts to diminish. Ultimately, if everything goes as expected, the patient's vision will improve, significantly reducing the risk of blindness. This makes ANXV a potential breakthrough solution for RVO patients all over the world.
So you saw what NXV can do for patients with RVO. I'm not telling you a little bit about the background of this idea. So Annexin A5 is a defense protein. It sits in every cell in the body. It's particularly enriched in barrier cells, which has an interface to the air or to the blood, etc. It's really in there to defend cells. So once there's a danger signal by the cells, it gets destroyed, gets disturbed, it flips on the outside of the cells, binds phosphatidylserine, which is the lipid that does the signaling, but Annexin A5 is sort of physically and practically binding to PS to reduce the signaling of PS. And therefore Annexin A5 has been called protective shield, band-aid, bloodhounds. It really homes into phosphatidylserine. And if you label Annexin A5 with a radioactive label, a short-lived one, and inject it into patients, you can actually see the joints of a patient with arthritis light up like this, where you can see the patient has undergone balloon catheterization in the artery of the leg, where Annexin A5 has been injected again. You can see it binds to the area which has been destroyed by the balloon catheter. And it has been used in up to 400 patients for this specific purpose as a diagnostic. With this overall very strong opportunities like protecting cells, you can select almost any indication which involves cell destruction and cell and inflammation. But we have focused on the niche indication called retinal vein occlusion. I will come back to that. But we have an interest potentially in all of the other ones listed on this image, including cancer and COVID-19. And in COVID-19 specifically, we have an interaction and collaboration with a Dutch team who wants to use our NXV in the clinical trial in COVID-19 patients. So when you do this, obviously you have to produce the compound under GMP conditions, as mentioned. We also have to do a phase 1 clinical trial. That's what we have done and reported. We do a traditional single administration dose and multiple administrations, so we treat healthy volunteers up to 5 days with the drug. And it says ongoing, but has been completed. We have no limiting safety issue reported. And this platform study will support any clinical development. Retinal vein occlusion is our focus. To the right, you can see how patients would experience the vision on one of the eye affected. It looks very dim. It can be completely blind, or it may be slightly less than this. But the cause is really a blood clot in the vein of the eye. The veins are supposed to lead the blood away from the eye, but if it stops, you get a back flow, and it gets swelling and destruction of the cells in the retina. Current therapies look like this. Antibodies are injected into the eye. It's the second most common cause of blindness. Actually, 16 patients diagnosed with this. So these patients will receive these treatment up to 6 times monthly. Once a month for 6 months. They call Lucentis, Eylea, and off-label Avastin. It's not really an effective drug. Only 30% get really improved. But still, it's a $20 billion plus indication. Its estimated value in 2023. So it's a huge market. Why we think NXV can work is that if you take out the red blood cells of these patients and bring them into a lab, you can see they are sticky to each other, and they will stick to the endothelial cells, which are coming from the macrovascular system. And Annexin A5 is able to reduce that stickiness and stop that adhesion to the vasculature. So for retinal vein occlusion, this can become a paradigm shift. We want to treat the patients early. We don't want to treat the same kind of mechanism or late-stage effects that anti-VEGFs would treat. Coming in early, we want to potentially unblock the occlusion. We want to protect cells at risk, but they're still viable in the retina, which are key to transfer the light into an image back in the brain. It's also an anti-inflammatory. We will have effects on vision. We'll have less causing of new blood vessel growth, which is what anti-VEGFs treat essentially. So there will be fewer anti-VEGF injections, meaning that we will tap into a huge market. We'll do an imaging trial in the Netherlands. I mentioned that. We hope to see that with the fluorescent Annexin A5 injected into patients with the disease, we will find that it binds to the right places in the eye, near the occlusion, near where the injury is. That study is supposedly starting any day now. So we have met with key authorities, the FDA, the British Authority, and the Icelandic Authority, where the leading person in the European ophthalmology field is working. We got a lot of good answers, but clear answers, and one of the reasons why we choose the US for our phase 2 trial was a good interaction with all the agencies, in particular with the FDA. So the phase 2 trial is really about studying the effect of Annexin A5, first and foremost for safety, because it's the first time in patients, but also for efficacy. We will explore multiple efficacy, not just vision. We will look into the eye with sophisticated equipment and see the survival of cells in the eye and look at blood flow, etc. About 20 patients will be investigated, and we are sort of at the brink of filing the IND to the FDA. So the company's strategy is really to have a very broad thinking about Annexin A5 and NXV. But we are focusing on the first in-human study, which we finished, focus on the imaging to support the phase 2 trial. We have got the regulatory advice. We will start the phase 2 trial hopefully late this year or early next year. And then we will sort of also work to find new indications. We're doing some explorative work in collaboration with the Dutch doctors. We're looking at COVID-19, but we think that we can actually expand into new territories during the next period of time. But first and foremost, RVO is our focus. Once we're done and hopefully got the positive phase 2 trial data, we're open to continue to develop this into the market, and we know from the discussions with authorities how to move forward. Should we find a licensing partner and we can get a good deal, we can do that as well. And again, we're working with the basic biology like NXV, which is a protective but naturally occurring protein, which we think we can take into many different directions. But our first and foremost focus is RVO. And with that, thank you very much for your attention.
Thank you so much, Anders, for this presentation. That's very interesting. So I just have a couple of questions for you. You just mentioned your clinical strategy for ANXV, and you're going into retinal vein occlusion there. Why did you choose retinal vein occlusion for your phase 2 study?
So there's a number of reasons. First and foremost, you have to look at the scientific rationale, where there was this, I think, the specific data coming from patients' red blood cells where they are sticky, and we can unblock that stickiness to them. So it's not only animal data, it's really human data, almost not clinical, but ex vivo data, as we call it. That was a strong rationale. It's also a disease where the clinical development program is not huge. It's for a company our size, we can potentially take it to the market ourselves. And also, it's amenable to really, you can look into the eye, you can do a lot of interesting experimentation. We do something different than the anti-VEGFs. So we're sort of working carefully towards that. And it's a huge market, even though most of the viewers or listeners have not heard about it. It's a $20 billion market. So if you just get 1.5% of that market, we're sell for a lot of money.
Yeah, absolutely. You also mentioned that the study will take place in the US. Why did you choose the US for this study?
Well, a couple of reasons. Obviously, the US is the largest market for the existing products as well. It's having the FDA's feedback and having the FDA approve your study and being involved in the development is a strength, no matter where you go in the end, if you go to the market on your own or if you talk to licensed partners. They will be really interested in what FDA has to say. And we had a good interaction with them in springtime this year, and I think it was a natural choice.
Mm-hmm. And finally, what would you say to potential investors who should be investing in Annexin?
Well, we have taken a lot of costs to produce the protein, which we have now at GMP quality. We have gone through the toxicology testing in animals. We've done the phase 1. So now many doors are open, like I said. So it's now the time when we're going to get clinical data, and companies like ours will, potentially get a much better and interesting valuation at the time you get clinical data.
Mm-hmm. Well, great. Thanks so much for answering the questions, and thanks again for your presentation. And we wish you all the best for your coming work.
Thank you very much.
If you look at Nasdaq's pipeline of upcoming IPOs within the life science sector specifically, do you think is there something special that the investors should keep an extra keen eye for going forward?
Yeah, of course you can see that there's been a lot of government aid in the society during the pandemic, and that is very good for many of the biotech companies also, because there's been a lot of capital out there to support the ecosystem. So just to give a number, the aid that have been given this time is more or less the same than the aid was given by the governments after the Second World War. So it's a lot of money. It's difficult to compare, but it is huge money, and these money are much more higher numbers than the dip that the industrial companies have done over the last year. So there will be a lot of capital available for biotech companies. So, but as I said, and I've said it many times, not only here, but also in the newspapers, the fourth quarter pipeline looks very promising. And I expect the IPO trend to follow the overall economy. And we don't see any hiccups in the economy for the next year. We will, however, see weeks with higher volatility than we're used to. And we have just seen one now where the share market did a small correction to the general high valuation we have seen in the market for the last couple of years. And in COVID, whenever you can do it, it's normally risky to wait to the last minute for these companies to go public, or to wait for their next value trigger to maximize the value of the company when they have to do a second round of a capital raise, because the market can sometimes be a little bit more tricky than we have used to see. So you need to move fast when you have the need for capital, and maybe, as we have had 10 years on a rally with only going up with the share prices, we will see some times the next year with a more flat market, and also maybe a more higher valuation, but the investors will be there. So I'm really confident that we can support our pipeline of IPOs.
We'll be looking forward to following the new companies coming into the market, of course. I would like to say thank you very much to you, Carsten, for taking the time to speak to us today.
Always, it's a pleasure to be there. Thank you, Jonas.
Next up, we have Gabather, and presenting for the company is CEO Michael Robin Witt. Welcome, Michael.
Thank you. All right, so I'll go ahead and give a spotlight, a short update on where we are at Gabather and this BioStock presentation. Thank you very much for the introduction. And I think I'll just go straight ahead with our first slide. And Gabather, we work a lot with cognition. Cognition is a key feature in mental disorders. And we're developing new therapeutic alternatives to treat deficits in cognition. And what cognition is in general is the way we experience and respond to the world. So if we see a situation like on this slide, through experience, we recognize the world, we have an emotional response to it, and a cognitive response to it, and react to the world. If cognition and the cognitive process is in a deficit or has some pathology, then our reaction to the world will be different because we experience the world differently, and then we have a mental health issue, a mental health disorder. It is the abundance of indications that are very interesting in looking at cognition in particular in mental health. So this can be disorders involving everything from autism to schizophrenia. Cognitive deficit are more severe in some disorders than in others, of course. Perhaps the most severe and most known is, of course, Alzheimer's disease. But it's a recurrent indication, a recurrent deficit in many mental health issues. And here we have a table, for example, where we basically tabulated mental health issues on the top row. And if you look in highlighted in red, they are all the symptoms that are relating to cognition in the different disorders. And you can see, just to repeat what I already said before, that cognition is a symptom and a deficit in the vast majority, if not all, of mental health issues. And of course, cognition and cognitive deficit is even a part of so-called healthy aging. As we all know, as we get older, both learning and memory become, if not impaired, at least deficit. So what are we trying to improve? What are we trying to get at? What are we trying to solve? The current treatments available for mental health disorders, and now it's a very general approach, very broad approach, have a number of advantages, but they certainly also have a number of disadvantages. And that can be anything from a simple failure to respond to the treatment, i.e. They simply don't work in a number of patients, although they have the same disorder, or they take a very long time to have an effect, or they have severe to very severe side effects, at least some of them, of those disorders. So there's a lot one can improve with new therapeutic alternatives like we're developing at Gabather. And at Gabather, we focus on, that's where the name comes from, GABA Therapeutics, and GABA is an acronym, stands for gamma-aminobutyric acid, long word. It means it's the key inhibitory neurotransmitter in the brain, and the brain, in a very simple analogy, is driven by both acceleration and braking. So GABA would be the inhibitory, the braking system in the brain, and glutamate would be the excitatory, the driving system in the brain. And it's the balance between excitation and inhibition that actually leads to healthy functioning. Is there an imbalance there, then you will have some deficit, or you will have an issue with mental health. For example, this is not to say that there are many other neurotransmitters who are very important in other disorders, of course, which are here a little bit on the side. I mean, dopamine is key, of course, and so are the other neurotransmitters. But we focus on GABA. We develop drugs that focus on the GABA-A receptor system in the brain. And we believe that the GABA-A receptor, being so important, is involved in either as a cause or an effect of mental health disorders in general. So it's a very interesting and very much used target also for therapeutic alternatives, ranging from anything from anxiety, insomnia, anesthetics, and so on. They all work on the GABA-A receptor, and our drugs work on this receptor systems in a new manner. So what we intend to do, to go back to the very first slide, if you look in the left-hand slide, we have an impaired way of experiencing the world, an impaired cognition. And this is what we're trying to solve, address, readdress, balance out again with our lead compound, in this case GT002, which is in the clinic right now. And as you can see, once you readdress the cognitive and rebalance the cognitive processes in the brain, then you will have, again, a healthy reaction to the environment and improvement in your mental health and mental health processes. The market for mental health therapeutic alternatives is large. It's increasing actually in correlating with an aging symptom. It is also increasing due to, for example, COVID, which we have seen in the last 2 years. COVID definitely affects the brain. And we all know that COVID has a large number of psychosocial and also mental health effects, negative ones, unfortunately, mainly. So the number of companies are developing therapeutic agents in the same area as Gabather, targeting the GABA A receptor, the number of deals, which are our potential partners in the future, which are large pharma. And the biggest one in this particular arena, so to speak, is a deal by Sage Therapeutics in the States, who's developed a particular neurotherapeutic agent for depression, and they licensed out to Biogen in a $1 billion deal last year in November. So that's the potential that is in this kind of therapeutics, in this kind of therapeutics arena, where Gabather is actually a player and wants to actually solidify our position also as a player and interesting partner in the future. So here we have our key compound that we developed over the last couple of years. This GT002. This is a compound that we developed from the lab bench to the clinic with our own proprietary model. We know how to make those molecules. We know how to synthesize these kind of therapeutic agents. And there's a number of very interesting effects in preclinical models. And many times I'm asked, how does it really work? How do you measure memory? And my favorite model is only one of them where we have seen effects of GT002. It's a so-called novel object recognition. And what you see in the boxes on the right-hand side, you use the natural curiosity of an animal. It investigates two objects that you put in the box together with it. And then you exchange one object for the other one. And then the animal knows, I already looked at the old object. It just learned it. I would investigate the novel object much more longer, much more thoroughly. And all you do is measure the time. How much time does the animal spend with the old and the new object? And you know that the animal has remembered. And using the curiosity of the natural animal, you can measure actually a learning process. This is just one model. I think it's a very nice one, but it also shows you how difficult it is within neuropsychiatry in general to generate models, which you then actually can translate eventually to cognitive deficits in man. But this is the first step. It's needed. It's very interesting. And there are not very many compounds that can actually do this kind of effect. So for GT002, we've developed GT002 as a lead compound in order to show that our compounds are efficacious clinically, not only preclinically. A couple of years ago, we transited from preclinical to clinical. And we've gone through actually 2 clinical trials in healthy volunteers, both single and multiple ascending doses. We've patented the formulation of our drug. It's actually being revised in the US right now. Our patent has been published. I think it's very nice. And we are now going into a target engagement study with GT002, looking at effect of EEG and fMRI, which I'm going to talk a little bit more later. Very encouraging results in the clinic. There's very good pharmacokinetic, i.e. how is the drug taken up, how is it metabolized, how is it excreted again, you want to get rid of again. It seems to be that we've been able to generate a formulation that can be given once daily in oral form, in a capsule form, which is of course an ideal formulation compared to, for example, some of the other formulations of competitor drugs, which are much more tricky to formulate and to administer as a pharmaceutical. So this is very convenient. We have synthesized and prepared a lot of clinical trial material at GLP level actually for future clinical trials. So we're ready with that. The next step in our program is of course the target engagement study. We will look at the effect of GT002 given orally to healthy volunteers. We will look at the electroencephalogram in the brain. We will look at the activity and the modulation of the electrical activity in the brain, which is the cartoon on the top right-hand side. And we will also look at where in the brain does GT002 modulate activity, which regions of the brain. And there we are, of course, interested in those regions of the brain, for example, that are presumed to be involved or have been suggested to involved in partially even proven to be involved in cognition. So that will be very interesting for us. The study is absolutely pivotal and a keystone study, because those data will then also later be able to guide us in the way into what indications are most relevant and perhaps most interesting for this particular kind of treatment. And we will also use this to benchmark against current treatments. So EEG is for us the key marker, the activity, and the EEG modulation is of course also relevant and interesting in a number of, if not all of other psychiatric and also neurological indications. So we're going to look at the effect of GT002 on the EEG, and from there then look at which indications could be most interesting. We have a number of candidates, of course, already, and we're going to benchmark it into together with, or against rather, current treatments. And this is our pipeline. So the key thing here is that we do still have a pipeline of other compounds. We have 2 patent groups we're working with. GT002 is the most advanced, most progressed one with the clinical trials and the upcoming target engagement study. But this is timeline just to show that, of course, since we can generate our own compounds, we still would generate new candidate drugs in the next year, or actually hopefully years towards the clinic, which is right now actually the territory of GT002 alone. Hopefully we'll have other candidates to make them even more interesting for example, partnering. So that is the idea, and that is the GT00X. So it's a compounds that are in the pipeline, which I actually want to highlight. Once we have then finalized the target engagement study and we have that data in, there will be a huge amount of data, very interesting one, then we can move on into phase 2 and partnering discussions in a number of indications indicated here on the 3 different colored lines following the target engagement study. And the reason that this is a rather lopsided slide is because we have put a lot of effort into GT002, because what's important to show that our therapeutic agents, our candidate drugs actually can be used clinically and not only at the lab level or the so-called molecular tool level. These are clinical candidates and very relevant ones. And we're getting quite a lot of interest since we have shown that it's a clinically interesting compound from both potential partners, but also academic groups that want to take a look. So this is next to last slide, where we are going with Gabather, and of course what we're looking at is partnering with GT002 after the target engagement, and we're already starting some discussions with it. And again, we have a pipeline of compounds coming up, and the idea is of course for GT002, novel approach, minimize the side effects of current treatments, and have a new therapeutic alternative within mental health and mental health disorders. Finally, this is of course a team effort. The top row is the management, and bottom row is the board of directors. I think a very experienced, very knowledgeable team that has driven this project forward on, I believe, very little small resources compared to what else you can use. And of course, one of the board members passed away 2 weeks ago, Morgan Sneedson. He's down here in the bottom row, and I would just like to highlight him because he was one of the founders of Gabather, and he's unfortunately not with us any longer. But this being a team effect, of course, we continue working with Gabather, also fulfilling his dream. And I think I have the last slide, and I think I've tried to keep within my allotted time, and so I have done. And I rest my case.
Thank you so much for that presentation, Michael. It was very clear and concise, and you talked about the target engagement study and why that's a key part of your clinical development with GT002. Could you go into a little more detail about that?
Yeah, right. Target engagement is absolutely key. It's actually a kind of study that has been requested, asked about by potential partners. What you want to see is, does the drug go into the brain? Does it modulate the activity in the brain? Where in the brain does it modulate the activity? And is that modulation different to what you already have as therapeutic alternatives? And this, once you have answered those questions, then you are very far in generating even more interest in potential partners. So target engagement, I've always said it's an absolute keystone, an absolute milestone for us And the amount of data that we're going to is going to be very huge and very interesting, actually.
Mm-hmm. And you've recently submitted a request for approval for the study to the Portuguese authorities. When might you expect that approval?
Difficult to divine bureaucracies all over the world, including Portugal. We can say that we submitted all our data. They're being processed right now. There might be questions. If there are questions, there's no question we can't answer. So we're very confident it's going to be soon. And as soon as it's there, we'll communicate it.
Mm-hmm. Also, you've recently talked about strengthening your management team and recruiting a new CEO. Why is this the right time for this kind of move?
Well, it's the right time for two reasons. The first one is that now that GT002 will go into a target engagement study, it will be out of the hands of any preclinical scientist like myself. So this will be run by itself. And we will have to focus on our pipeline. And that is where my strength is, my expertise comes into Gabather in the best way. And I think it's a very good idea to get somebody with experience and network in the investment community, in the investor relation community, and me to focus what I'm best at. And I'm a scientist by training. I started as a scientist, Gabather, to go back to the lab, basically, and I look forward to it.
Mm-hmm. Well, speaking of structure of the company, you also touched upon the passing of your co-founder and board member, Mogens Nielsen. That's obviously a huge loss for the company. How does it affect Gabather?
Yeah, again, thanks for the question. Yeah, it happened 2 weeks ago, very sudden. Mogens has been a founder of Gabather. Many of his ideas of the model that we work in come from him, his lab, including, of course, Olof Sterner's lab in Lund, and myself. I also worked there. So, since we do this as a team, all this is, of course, salvaged towards the next step for Gabather. And I worked together with Mogens for 36 years. And I think the best way to honor him and to thank him is to get GT002 or any other compounds from Gabather into patients, which was his big dream. And I think it's the dream of any preclinical scientist working in drug discovery to see a compound from the lab bench actually into the patient, and hopefully with a good effect for the patients. So that would be the very best way of saying thank you to Mogens.
Mm-hmm. Well, thank you so much for answering these questions, Michael, and we do wish you all the best in your
future work. Thank you very much. Thank you very much for having me.
The next company to present here from the Lund studio is Ectin Research, which will be presented by CEO Anna Sjöblom-Hellén. Welcome, Anna.
Thank you. So my name is Anna Sjöblom-Hellén. I'm the CEO for Ectin Research. Ectin develops cancer therapy against metastasizing urethral bladder cancer. The company is situated in AstraZeneca's BioVenture hub in Mölndal. The company was also very recently listed on a spotlight stock market. Short about the background behind the company. One of the founders, senior physician Christer Edlund, he had a patient with a primary tumor which was a muscle invasive form of urethral bladder cancer. The man got standard treatment for his disease, but unfortunately 6 months later the cancer had come back and now it was metastasizing to the pelvic area, it was potentially in the lungs, and he could not get any further treatment. He was unfortunately expected to live just a couple of months more. On top of everything else, it was found that he had a parasitic infection, which he got treated for. 3 months later, the man came back feeling very well. This was really astounding to Christer, who did not expect it. He was examined again and it was found that the patient was totally relieved from cancer. So they looked into the previous samples that they had collected and they could see that he had suffered from a really severe form of urethral cancer. And that was totally clear now. So it's based on this that we are now developing something which we call the MFA 370 to treat metastasizing urethral bladder cancer. Short about bladder cancer, it's actually the 5th most common cancer in the US and Europe. Just in the US, the healthcare costs 2020 was $173 billion US. And if you look into the 5-year survival for patients with metastasizing urethral bladder cancer, it's just 5% currently. And yearly, 200,000 people die from bladder cancer every year. So it's clearly really high unmet need currently. So current treatments have very limited efficacy when treating metastasizing urethral bladder cancer. And it's also associated with severe side effects, giving these poor patients very poor quality of life. And it's also associated with very frequent and long hospital visits. And that gives the society very, very large costs. And it's actually the most expensive cancer to treat today. So, regarding treatments, when you treat metastatic urethral bladder cancer in Europe or US, this is what you usually do. You start off by giving the patient as a first-line treatment, if they are well enough, something called platinum-based chemotherapy. And if that doesn't work, you move on to second-line therapy, which is a checkpoint inhibitors. And then if that doesn't work, which it usually doesn't, you move on to third-line treatment, and that is present in the US, something called an antibody-drug conjugates, Padcev or Trodelvy. And it's here that we would like to enter with our MFA 370. There are subgroups of patients, patients who are too sick to get platinum-based chemotherapy, and they get checkpoint inhibitors as their first line of treatment. And then, as a second-line treatment in the US, the patient gets Padcev if the first didn't work. And then there are subgroups of patients having specific mutations in their tumors. And then they can get something called the FGFR kinase inhibitors, Balversa. And that is then only in the US. So we would like to enter as a third-line option for these patients. And then after approval, go for the lower lines. So how many patients are we talking about having metastasizing bladder cancer in the US and the 5 largest European countries? So if we look on 2020, the total number is 58,100 patients to be treatable with first, second or third line treatment. The third line represents 8,100 patients. This is numbers that will forecasted to increase with about 22% to 2030. Looking into the market potential in the same area, the total market is $4.23 billion US, and that is first, second and third all together. And that is based on the cost for Padcev in the US and forecasted numbers for Europe. For third-line treatment, that would give us $616 million or 769 2030. And then if we go to MFA 370, which we believe is a risk-reduced project, it all started, as I mentioned, with the clinical observation that one of the founders made. A lot of research has gone into this project, and they are now 3 founders, and they established something called the MFA 370. And MFA 370 is actually 2 different molecules, and those 2 molecules are used today but in totally different indications. So what we are doing is we are repositioning those 2 substances in order to treat metastasizing urethral bladder cancer patients. And since these molecules are known, it's very much information already established out there that we could use. It's toxicology studies, there is different kinds of lab studies that have been done where one has seen that both of these substances have anti-cancer effect. So the Swedish MPA has actually now granted us to go into the next step with our project, a clinical development. If we then look into MFA 370's different advantages, we will give this treatment as an oral administration, so the patient can get this treatment at home. And they can also get it at home due to the fact that these 2 substances have excellent safety profiles. That is known from before. Together, it will give reduced societal costs, something that is important today. It will be even more important some years from now. And these next thing with scientifically well established, I've mentioned this before, these 2 substances have been studied both in vitro in the cell lab as well as in humans. So much information is already out there. And regarding this being a repositioning project, this will give us a reduced developmental time and also, very importantly, cost, and nevertheless also a decreased risk going forward. So what you see here is some preclinical data supporting the clinical trial application that we filed to the CTA, MPA, I mean. What you see is a cell proliferation experiment using urethral bladder cancer cells. To the left, in gray, you see the control. That cell number is put to 100%. If you add a small amount of one of the components in MFA 370, an NSAID, you see that nothing happens. If you add a small amount of the other component, an ivermectin, nothing happens. But if you start combining the 2 in very small quantities, you get a strong synergistic anti-proliferative effect, an anti-cancer effect. And if you start looking at these cells in the microscope, what you see there is that you will see that they have gotten a signal that they should die. And this is exactly what we think happened to the patient. That is, apart from stopping, the tumor he had did not just stop growing, it actually vanished and disappeared. So here you have the plan for our clinical validation. This is a plan that we co-developed with our CRO, Link Medical Research, and it's part of the clinical trial application that we sent into the Swedish Medical Product Agency. It's rather recently was approved by this phase 1 part, which we will do on patients with metastasizing urethral bladder cancer. So 10 patients will be studied, and the primary endpoint we are looking at is safety and tolerability in these patients. What we will also establish is something called recommended phase 2 dose. And that is the dose we think will give us maximum clinical effect in the phase 2 part of the clinical trial. The phase 1 part will be initiated late this year, and the result is expected late next year, 2022. And this result will then be reported to the Swedish MPA, and they will then decide whether we can start the phase 2 part. The phase 2 part is then a clinical proof of concept study, and what that means is that we will statistically show that MFA 370 has clinical effects in these patients. And we are now talking about an additional number of patients being studied. Also here, patients with metastasized urethral-bladder cancer. The study is planned to be initiated H1 2023, and most of the data will be generated H2 2024. The primary endpoint I should mention is something called the overall response rate. And we are very happy to announce that due to the listing of the company, the phase 1 part of the clinical trial is fully financed. And regarding patent status, we have today approved and issued patent in US, Europe, Australia and Canada. The patent will lapse 2037 with 5 years patent extension, which you usually get. We have also examined something called freedom to operate, and that has also been confirmed. So we know that we can use this patent when we do a commercial development. New potential IP. By doing this clinical trial, we will generate a vast amount of information. One thing that could be patentable is the dosing regime to achieve clinical effect. Another thing that could be patentable is that if we find some biomarkers that we could stratify patients in responders and perhaps non-responders. And finally, one very important patent that we would like to do is to do a formulation patent, and that is something we would like to combine the two substances in something called a fixed dose combination. And this fixed dose combination, the thought with that is to maximize the effect these two substances have when they come into man, and also facilitate for the patients to do the correct dosing to achieve maximum clinical effect. Another thing that we see as potentially possible is something called orphan drug designation in the US. And that would facilitate product development, but even more important, it would increase market exclusivity. And then we have data protection or registration, 11 years. That would give us a product protection until 2040. Here you see the management and board of directors for Ectin. In the management, apart from myself, we have Chief Medical Officer Stephen Glaser. He's a medical doctor. He has worked as Chief Medical Officer in a number of different companies. He's very focused on oncology and oncology trials. We have Chief Financial Officer Michael Owens. And Michael has also worked in listed companies and done a lot of financial reporting for them. And then going to the board of directors, they are headed by Fredrik Sjövall, who is the chairman, and he is also heading other listed companies currently, such as Monivent, Lipidor och Sicum. And then in the center, you see the 3 founders of the company and also inventors of MFA 370. We have the senior physician who had the first clinical observation. And then we have Professor Emerita Marie-Louise Ivarsson and Associate Professor Peter Falk. And then to the right you have Carlos de Sousa, who is industry professional. He has been working within the life science industry now for more than 30 years. He is currently the CEO for Ultimovacs in Oslo. Then we have Fredrik Andersson. He is a private investor and entrepreneur. And finally, Anders Vaz, who is an investor too, representing GU Ventures. So, we strongly believe that MFA 370 is a risk-reduced project for the treating metastasizing urethral bladder cancer. We have demonstrated clear positive results regarding the combination, and we also, depending that this is a repositioning project, have a very attractive product development timeline as well as cost ahead of us. And regarding Ectin's target market, I mentioned to you previously that it's really substantial, and unfortunately for these patients, however, the competition in the area is limited. So there is a high profitability potential for investors and also for potential acquirers. And with this, I would like to thank you.
Well, thank you for the presentation. Just have a couple of questions for you.
Yeah.
Like you said, Ectin has been listed since August now.
Yeah.
What is the most important thing that's happened to the company since then?
Yeah, it's definitely the clinical trial application that the Swedish MPA approved. because that has reduced the risk of developing MFA 370 further, going into man.
You mentioned here about your initial indication, but if we look beyond that, how do you see the potential in other indications?
Our preclinical data is really suggesting that we may use MFA 370 very beneficially also to treat patients with prostatic cancer, breast cancer, colorectal cancers. And so next year our goal is to, based on the scientific knowledge as well as whether we can make a business case out of it and the new cancer indication, then choose a second follow-up cancer indication to co-develop, hopefully, depending on finance of course, with our bladder cancer project.
And finally then, if we look at short-term goals, which are the most important ones for you?
The short term is to really sum up, all the regulatory approvals and getting that all together, so we are able to start up our clinical trial in Sweden. And then very importantly also to follow up, we are preparing additional CTAs in 2 European countries more. So in order to, increase the recruitment of patients for our phase 1/2 trial. And then lastly, of course, to decide on a second cancer indication to co-develop.
Well, it looks like you have a very exciting period ahead of you then.
Yes, we believe so, yes.
Thank you so much for coming here and telling us about it.
Thank you. Thank you.
Next up from our studio here in Stockholm, we have Sprint Bioscience, and presenting is CEO Erik Kienman. Welcome, Erik.
Thank you. Great to be here. So before I start presenting Sprint Bioscience, I'd just like to give you some numbers to think about. 2019, some 66,000 new cancer patients got the diagnosis in Sweden. The same year, 22,500 actually died from their cancer. On a global level, 2020 there were 19 million new cancer cases, and that is expected to grow to some 30 million by 2040. So clearly there's a great unmet medical need, a need to develop more effective and more safe cancer therapies. And this is what Sprint Bioscience is about. Our mission is to contribute to that, to deliver breakthrough preclinical oncology drug programs to licensed partners. And this will take us to our vision, which is to become a profitable growth company. This slide summarizes what we've achieved to date. We do have a multiple and increasing sources of revenue portfolio. We have a track record by now in terms of 3 out-licensed programs with a total revenue opportunity of $747 million and on top of that significant royalties as well. We have 2 programs in the cancer field. One is the Petra-01 program. The second is VPS-34. And then we also have a NASH program. In addition to this, in our portfolio, we have 3 internal cancer programs. That's VADA, DISA, and NEMA. And these are and constitute new licensing opportunities. In our very cost-effective, agile, and flexible organization, we can rapidly develop 1 new preclinical oncology program per year. Our focus is on scientifically validated new target proteins which fulfill the opportunity of an unmet medical need. And in addition to that, obviously, a commercial potential. Through our very agile process, we are able to develop programs that actually offer the potential license partner a first-in-class opportunity, which is important in this field. So altogether, we have multiple high potential revenue streams possibilities, and so far we've actually received some 158 million Swedish crowns, which have helped us in the development of the company and also the development of the portfolio. The slides summarizes our place in the drug development process. We are filling a gap in the industry in terms of providing new starting points for effective drugs. So we're focused on the early stages of development, and what we deliver is lead drug candidates with quite limited initial investments. We then out-license this completely to a partner who actually provides the further funding and the resources necessary to take the programs to the market. And we receive an upfront payment, and the deals will usually involve also milestone payments throughout development, and once on the market also sales milestones and significant royalties on sales. The slide summarizes the out-licensed programs in our portfolio. So we have the VPS-34 program, which addresses immune oncology, and I will get back to that in a moment. This is in preclinical development, being prepared to move into clinical development, and the license is to Deciphera, a major US oncology-focused program with 1 product on the market and several clinical programs already in their pipeline. The total deal value of this program is some $277 million. We then have the Petra-01 program, which is developed by Hybricell, another oncology-focused US company. This program addresses autophagy and cancer cell growth. Is also in the preclinical stage, being prepared for moving it into clinical development. The total deal value of this program is some $240 million, and also here there are significant royalties if there would be products on the market. In addition to this, we have a NASH program, which is an earlier stage program, still in preclinical development, together with LG Chem, a Korean pharma company. This is a summary of our internal programs. We have VADA, which is the most developed program in this part of the portfolio. This program addresses DNA repair and cell division of cancer cells. We are targeting partners and have several discussions ongoing when it comes to this program. The DISA program addresses also solid tumors and actually addresses a mechanism that cancer cells utilize to escape an immune response. We have just announced the target protein when it comes to this, and we'll start presenting the program to interested potential license partners, starting by Europe, which is a partnering meeting taking place at the end of October. Then we have the NEMA program, which is a new program we quite recently announced, which addresses several mechanisms involved in cancer survival and cancer growth, and also immune escape. And we will start targeting potential licensing partners with this program sometime next year. So this program, this slide, actually summarizes our long-term vision, namely to become a profitable growth company. So we have the 3 programs that are out-licensed already, and then we have the 3 internal programs, which are new license deal opportunities, and these together build multiple sources of revenue streams, and over time these will increase as the programs mature, and will get us to the goal of being profitable and cash flow positive. This is a high-level summary of our process, a very agile process, taking us from the idea, where the starting point is academia. So the good news when thinking about the problem of increasing incidences of cancer and still very high unmet medical needs within the space is that there are very novel scientific opportunities. And what we want to do is to industrialize these ideas. So when there is a target protein which addresses a mechanism which has the potential to actually fulfill an unmet medical need, we will make models of this protein from our fragment library. We will then, in an iterative process, develop molecules that can be the starting point of future drugs. And if this is all possible and we can validate that these actually act as expected in terms of the mechanism, these are then out-licensed. And this process takes, on average, some 1.5 years. This should be compared to the industry average, which is some 5 years from idea to molecule. Our focus areas involve several areas relevant to cancer survival and cancer growth. One is the immune system or immune response. Another is tumor metabolism. So cancer cells need a lot of energy to grow. And because they are cancer cells, they need to use other alternative ways of generating the energy needed. And these mechanisms can be addressed. Another is the microenvironment of the tumor. So because the tumor cells are growing very rapidly, they need certain mechanisms to actually enable that rapid growth. BPS34 is one such opportunity. So tumors have the problem used the mechanism of actually escaping the immune system by stopping immune cells from invading them. So the term for this is cold tumors. What this program does when inhibiting the protein VPS34 is that tumors again will be invaded by immune cells and activated immune cells. The tumors are then hot, which will actually enable the immune system to fight the cancer. The graph here shows an experimental model looking at melanoma cancer. The red line is the growth without any treatment. Then you have the yellow line, which is checkpoint inhibitors, a modern immune oncology type of treatment which stops some of the growth. Molecules from our program will have an even better effect, and combining these two will potentiate the effect of both these type of treatments. So the opportunity is both monotherapy but also combination therapy. And looking at the overall benefit these therapies actually introduce, it addresses only 13% of the market. So combination therapy will actually potentiate that commercial potential considerably. So the Petra-01 is also out-licensed. It addresses leukemia. The graph here shows a very potent effect on tumor growth. This is developed now further by Hybricel, VPS34 by Deciphera. We have the NASH program, which also is out-licensed. It's early stage. It addresses fatty liver, which is a huge commercial opportunity and also a high medical need since there are no treatments in this field yet. And what the protein does, it's involved in both fat uptake, liver inflammation, liver fibrosis, and liver fibrosis turning into cirrhosis and liver cancer. The program is now in preclinical development, and we are collaborating with the Korean company LG Chem in terms of the further development, preparing it for clinical development. So the VADA program is a mechanism that cancer cells use to actually repair faulty DNA, which often occurs in cancer cells because of their nature, and also assists in cell division and multiplying of cells. Turning this mechanism off will have the same type of effect as radiation therapy and chemotherapy. Since the cancer cells then have fragmented, damaged DNA, and also the cells can't divide, the immune system can also attack the tumor. This addresses the solid tumor market, a huge market, where colon cancer and pancreatic cancer is good examples. The DISA program is the program that we recently announced, a new program addressing Tregs, which is a mechanism that cancer cells use to escape the immune system. By blocking this, the DNA fragments in the cells will be recognized by the immune system, and the immune system can assist in fighting the cancer as well. The NEMA program addresses several mechanisms. So the target NEMA is used by cancer cells to actually assist in the tumor metabolism, generating the energy that cancer cells need, and also in setting up the microenvironment necessary for cancer cells to grow, and also escaping the immune system. By blocking this, the situation in the tissue will be normalized, and the cancer can be attacked. This protein is elevated in cancers with poor prognosis. Good examples of that is ovarian cancer and glioblastoma. So in summary, we have a compelling portfolio with multiple and increasing sources of revenue. We do have a track record of delivering high quality preclinical cancer programs, and by now we have 3 out-licensed programs with a total deal value of $747 million, and on top of that, significant royalty opportunities. We have 3 out-licensed, we have 3 internal preclinical programs which constitute new possibilities in terms of out-licensing deals, and we have the capacity to deliver with limited investments 1 additional program at least per year to add to the portfolio and giving us further opportunities to reach our long-term vision to become a profitable growth company. Well, thank you very much.
Thank you so much for that presentation, Eric. I just have some questions for you.
Sure.
I'd like to begin by the out-license programs that you mentioned. Since you've signed quite a few, how would you say these affect the interest in the company?
Well, as I said, I think we by now do have a track record. I think this validates the quality of what we're doing. So the sort of starting point in terms of the molecules and the series of molecules that we develop, and also in choosing the right protein, and also it validates the sort of the business model in taking them to the preclinical stage, and this is still quite early in development. And as we are actually striking deals, I think that this validates both the business model, our ability to strike deals with good partners. And as we've done this, I think that other companies are realizing that we do have the potential to deliver. And it actually has had the impact that there is growing interest, and people in our network are actually asking for meetings just to hear what we are actually doing rather than us approaching them.
All right.
Yeah.
That's great.
Yeah.
So talking about financing, right now you're quite well financed. What does that mean overall for the activity of the company?
Yes, this is very true. I think we're in a better situation than we've actually ever been in terms of the company history. So the director chair issue, the rights issue, the recent deal that we did with Decipher, and the convertible that we did all together has put us in a very favorable situation in terms of focusing on actually accelerating what we're doing in terms of delivering new projects, discussing projects with potential partners, and also interacting with the current partners, doing the best we can to assist them in taking these programs to the next milestone.
Mm-hmm. Recently Björn Höstrand was elected chairman of the board for Sprint Bioscience. How has his entry affected the company?
I think he brings with him a lot of good new things. We are, as I'm sure you're aware, a couple of people who are new. I myself, I joined the company some 8 months ago, and we have a new CFO. And Björn is new since this summer as the chairman of the board, and he has brought his leadership to the board. What he's also brought is a clear engagement in the company. It's not just that he's actually bought into the share and has skin in the game, but he's very engaged in what we're doing. And he contributes, I believe, very much with his international experience as an executive, being CEO of both Swedish companies but also international companies, and being very much involved in both M&As and BD activities around the world. So he understands this business, which is also very important for us.
Mm-hmm. That's great. Thank you so much for answering the questions, and thank you again for your presentation, and we wish you all the best with your work going forward.
Thank you very much.
The next featured company is Saniona, and here to present is CEO Rami Levine. Welcome, Rami.
Thank you, Michael, and thank you everyone for having us. And it's my pleasure to introduce you to Saniona. So who is Saniona? We are a clinical stage biopharmaceutical company. We're focused on discovering, developing, and at some point also commercializing our innovative treatments for rare diseases. We are publicly traded on NASDAQ in Stockholm. We have our research organization in Copenhagen and Denmark, and we have our corporate office in the Boston area. Our lead asset is Tesamet, which we are developing for 2 rare disorders. The first one is hypothalamic obesity, and the second one is Prader-Willi syndrome. In hypothalamic obesity, we conducted a phase 2 trial, which I will share with you a bit later. And we have a very clear path forward with the FDA on the continued regulatory and clinical development. Our next clinical trial for hypothalamic obesity is a phase 2b trial, which we intend to initiate before the end of this year. The second indication is Prader-Willi syndrome. Similarly, we conducted a phase 2 study. We have been in constant dialogue with the FDA, and our next trial will also be initiated before the end of this year, and it will also be a phase 2b trial. So 2 phase 2b trials with Tesamet will be initiated before year end. But we are not just about Tesamet. Yes, Tesamet is an asset that we are very excited about. It's in mid-stage clinical development, but we also have a drug discovery channel, which not every company obviously has. And what our ion channel drug discovery engine will do is it will fuel our future pipeline as we advance and build the company. The most advanced asset coming out from our ion channel drug discovery engine is SAN711, which we are exploring for rare neuropathic disorders. We started a phase 1 trial with SAN711 back in June, and we are expecting results from that phase 1 in the first half of next year. The next asset coming out from our drug discovery engine is SAN903, which is about a year behind SAN711. We are exploring SAN903 for rare inflammatory, fibrotic, and hematological disorders, and we are expecting to initiate phase 1 with SAN903 in the second half of next year. But behind these 2 assets, we have a whole library of over 20,000 ion channel modulators, which we intend to continue to explore, to continue to investigate with the intent and a very clear goal to identify the next treatment for the next rare diseases. The first 2 pillars that I just talked about, Tesamet on one hand and our ion channel drug discovery engine on the other hand, is really where the company's focus is moving forward. But historically, we have had a slightly different focus. Our focus was on developing and ultimately, or discovering and ultimately out-licensing our assets to other companies. While that is no longer our strategy, we do have out-licensing agreements in place from that strategy. And what that will do is, number 1, validate our science, but number 2, it will be a nice revenue stream coming into the company. It won't be our main focus, nor will it be our main revenue stream, but it will be a secondary revenue stream as we advance, or as the companies advance those assets and we get milestone payments and ultimately royalties if the products reach the market. One of those agreements is for CAD 1883 for movement disorders, which we originally out-licensed to Ectin Therapeutics. Back in January of this year, Ectin Therapeutics was acquired by Novartis, and we received an upfront payment of nearly $3 million for that. And as they continue to develop it, we will receive milestone payments and ultimately royalties if the product reaches the market. Another agreement that's in place is with Boehringer Ingelheim. This is for a preclinical asset for their schizophrenia program. And under that agreement as well, we will be receiving milestone payments throughout the development. And ultimately, if and when it reaches the market, we will also be receiving royalties. And last but not least, we have an out-licensing agreement for Tesofensine for general obesity. We out-licensed Tesofensine to a company called Medix, who have obesity clinics in Argentina and in Mexico. Medix conducted a phase 3 trial back in 2019 and submitted their file to the Mexican authorities for approval in December of that year. They are now waiting for feedback from the FDA, obviously, hopefully an approval. There were some questions late, just a few months ago, and they are now expecting to come back to the Mexican authorities and hope to get an approval early next year. Under that agreement as well, we will be getting double-digit royalties once the product is on the market. And last but not least, from a financing perspective, we have robust financing to drive our current operating plan. We are well funded into the second half of next year, and we are covered by some strong institutional support from RA Capital to Pontifax to New Leaf and others. As I mentioned, we have built our corporate office in the Boston area. This is our executive team. Almost everyone is based here in the US with their teams. The only one who's still based in Copenhagen is Jürgen Dreyer, who's our CSO and founder, and he leads our research team in Denmark. Our R&D pipeline is shown here. As I mentioned, we are advancing Tesomet for hypothalamic obesity and Prader-Willi syndrome into phase 2b. These trials will be initiated before year end. SAN711, which is a GABA alpha 3 PAM, which we are targeting for rare neuropathic disorders, is in phase 1, and results are expected in the first half of next year. SAN903, about a year or so behind SAN711. SAN903 is a calcium channel inhibitor, which we are targeting for rare inflammatory, fibrotic, and hematological disorders. And we are expecting SAN903 to move into the clinic in the second half of next year. And then obviously behind these assets, there's a whole database, as I mentioned, of over 20,000 proprietary compounds, which we intend to continue to investigate to try and identify our next treatment to move into preclinical and clinical development. But let's start and talk about Tesomet, obviously our lead asset moving into phase 2b. Tesomet is potentially first-in-class triple monoamine reuptake inhibitor. It is a fixed-dose combination of tesofensine and metoprolol, and it is designed to reduce hyperphagia, which is one of the main characteristics seen in both hypothalamic obesity and Prader-Willi syndrome. And it does it by targeting the reward center in the brain, increasing the levels of dopamine and serotonin in the reward center, and by doing so, reducing the craving for food and reducing the appetite. Tesomet also targets the appetite center in the brain, and by such increases the level of noradrenaline in the brain. And by increasing the low levels of noradrenaline, it also increases the metabolic fat burn, all of which are critical for the two diseases that we are talking about. So let's go into a bit more detail into these two therapeutic areas. The first one is hypothalamic obesity. Hypothalamic obesity is an acquired rare disease. Most commonly, it's caused by damage to the hypothalamus in the brain, and most often that happens after the removal of a rare brain tumor called craniopharyngioma. These are typically healthy people who suddenly had a rare brain tumor, and as a result of the removal of that brain tumor, the hypothalamus in the brain is injured. And as a result of that injury to the hypothalamus in the brain, there is a rapid and excessive weight gain post hypothalamic injury. These patients could be normal, healthy, even very active athletes, and within a year post-surgery, they become obese. Hyperphagia, which is this uncontrolled hunger. Many of these patients, their parents have to lock the fridges, have to lock the cabinets. This is probably the most difficult aspect of the disease, is to manage what these children eat. Some of the other characteristics are memory impairment, attention deficit, and impulse control. There's currently no FDA-approved drug for hypothalamic obesity. In fact, Tesamet is the most advanced in clinical development for hypothalamic obesity. In terms of the size of the patient population, in the US, there are between 10 to 25,000 hypothalamic obesity patients. In Europe, between 16 and 40,000. And here on the right, you can see an example of Ali, who is living with hypothalamic obesity before and after surgery. On the left, you can see her before surgery, and on the right, this is literally less than a year after surgery, and you can see the differences. So we conducted a phase 2 trial. In this trial, we saw not only did we meet some primary and secondary endpoints on efficacy, but also we tracked obviously safety, and the product was effective and safe. What we saw was about 6.3% reduction in body weight versus placebo. This was significant and clinically meaningful. The FDA came out with guidelines on weight management. Basically, anything above 5% is considered clinically meaningful. We were able to show 6.3 or nearly 6.3% reduction in body weight, which was also statistically significant. We also were able to show that 61% of the patient had over 5% of body weight loss. So this was not an outlier. When you look at that criteria from the FDA of at least 5% body weight loss, 61% of the patients on Tesamet we were able to see that weight loss. The other parameter that the FDA looks at is glycemic control. In this study, there were 2 diabetic patients, and both of those patients were on Tesamet, and what we were able to see is that nearly 49%, there was a 49% lowering of the HbA1c in those 2 diabetic patients. So not only body weight loss, but also glycemic control was under control. So from an efficacy perspective, very promising. We obviously also looked at safety, and Tesamet was generally well tolerated in the study. So with that, we met with the FDA and have been discussing with the FDA what the next stage of clinical development will be. And the next stage, as agreed with the FDA, is a phase 2b clinical trial. This will be a much larger clinical trial. In the phase 2, there were 21 patients. In this study, we're targeting about 110 patients in the study. It will be double-blind, randomized, placebo-controlled. You can see there will be a placebo group and 3 different doses of Tesamet. We are planning sites globally, both in the US and Europe and other countries. Primary endpoint will be change in body weight as per guidance from the FDA. This will be a 30-week study. After the 30-week study, there will be a rollover into an open label extension of another 24 weeks at the highest tolerated dose. We are expecting data from the HO phase 2b trial in the second half of 2023. Let's talk a bit about Prader-Willi syndrome. Unlike hypothalamic obesity, Prader-Willi syndrome is actually a genetic disorder, and that is caused due to the absence of a defective parentally expressed gene on chromosome 15. So these are typically patients who have hyperphagia is the most concerning and characteristic of the disease. These are patients who typically have abnormal growth, abnormal body compensation. Unlike in hypothalamic obesity, as I mentioned earlier, these were healthy patients. These are unhealthy patients to begin with. Their body composition is very different to a normal healthy person. They have very low muscle tone. They have social, emotional, and cognitive deficits. They have obesity-related comorbidities, and there currently is no approved treatment for PWS, specifically no treatment for the hyperphagia of PWS. This is a slightly larger indication than hypothalamic obesity, but just ever so slightly. There are about 11,000 to 34,000 patients in the US and about 17,000 to 50,000 in Europe. And here on the right, you can see a few PWS or people living with PWS at a home for adults living with PWS. The phase 2 study that we conducted was a 2-part study. The first part was in adults for 3 months versus Tesomet versus placebo. And the second part was in adolescent patient, where the first part of that study was Tesomet versus placebo, and then open label extension for another 3 months, and then a second open label extension for another 3 months. And what you can see here, the first 2 bubbles are the adult patients and the results from those. Reduction in body weight was seen at 5.4%, which again was clinically meaningful. And when looking at the hyperphagia, which in PWS is the main primary endpoint in the future clinical study, we saw an 8.1 point reduction in hyperphagia, which is highly statistically significant and clinically meaningful. When looking at the adolescents patients, we saw a reduction in hyperphagia and body weight only when we increased the dose to the higher dose in the second open label extension at a dose of 0.25. The adults were at a dose of 0.5. And last but not least, we obviously also looked at safety, and Tesomet was generally well tolerated in this study as well. And with this data, we have been in dialogue and discussion with the FDA. We have a clear path forward and alignment on the clinical trial, which will be a phase 2b, which will start before the end of the year. This will be a 16-week double-blind study conducted in US, Europe, and other countries that you can see here. The primary endpoint will be hyperphagia, and there will be 3 doses of Tesomet versus placebo, a 0.125, 0.25, and 0.375. And after the 16-week double-blind phase of the study, the patients will roll over into an open-label extension of 36 weeks at the highest tolerated dose of Tesomet. Top-line results from the PWS trial are expected in the first half of 2023. So in summary, some of the key milestones that we have achieved so far and are expecting over the next couple of years. So we received earlier this year orphan drug designation for Tesomet in Prader-Willi syndrome. We have a clear path forward for Tesomet in hypothalamic obesity following the conversations we have had with the FDA. Both of those were achieved in Q1. In Q2, we initiated our phase 1 trial for SAN711 in healthy volunteers. That was in June. And in Q3, we received orphan drug designation for Tesomet in hypothalamic obesity as well. Moving forward, we will be initiating 2 phase 2b trials, as we just discussed, both in hypothalamic obesity and Prader-Willi syndrome. Both of those will take place before year end. Top-line data from the phase 1 clinical trial of SAN711, which is underway, is expected in the first half of next year. And then we expect to initiate another clinical trial, a phase 1 trial with SAN903 in the second half of next year. And then as we move into 23, we're expecting to start receiving the top-line results from our 2 phase 2b trials. During the first half of 23 will be the results from Tesomet in Prader-Willi syndrome. And during the second half of 23, we will be receiving the results from Tesomet in hypothalamic obesity. So this is just a character to try and illustrate that for us, Tesomet, even though obviously the most advanced asset that we have in clinical development, for us, Tesomet is really only the beginning as we prepare and plan to grow the company. And you can see below, obviously, Tesomet and HO and PWS is the starting point. We have started SAN711 in phase 1. Our goal will be to continue to develop that. And SAN903 is about a year behind. We plan to bring that into clinical development as well. Behind these assets, we have a whole library which we'll constantly continue to explore with the intent to identify the next treatment for a next rare disease. We will also be looking at life cycle management. Beyond the lead indications that we are currently pursuing, are there other indications where the treatments that we are working on could be beneficial? Then conduct the appropriate clinical trials and bring those to market as well. And then in parallel to all of this business development, we just announced the hiring of a new chief business officer. And her role will be to explore a variety of different business development opportunities for us to either partner, out-license, or in-license as we continue to build the company. And with that, I'd like to thank everyone for your attention. Thank you.
Well, thanks so much for your presentation, Rami. It seems like Saniona has a lot of exciting things going on, and but I'd like to ask you a little bit about the share price, which has been slightly on the decline in recent months. What would you say is needed to improve it?
That's a good question, but I mean, as you know, we are a publicly traded company, and I can't really comment on the share price. We are not happy, obviously, with the decline, and we don't believe it reflects the progress that the company has made. What I can say is that drug development is a long-term endeavor, and progress is not always reflected in the day-to-day share price, as we can see. And we have made a lot of progress. As I mentioned earlier, we are advancing Tesamet into 2 phase 2b trials, which will be initiated imminently, both for PWS and HO. We've initiated phase 1 trial for SAN711. We've received orphan drug designation for both hypothalamic obesity and PWS. So clearly, the company has advanced on the clinical and regulatory side as we have committed. And we're very excited about that. The share price is not always reflected in that progress. But we do strongly believe that if we continue to execute on our long-term strategy, which is bringing these assets to the market, to the patients who desperately need them, the share price should follow over time.
Mm-hmm. Well, along that note, you mentioned a little bit about what's coming next for Saniona. What would you say will be the most will be driving value the most in the future?
It's a great question. So obviously we have quite a few milestones. Obviously starting the phase 2b trials will be critical. The data coming from those trials expected in 2023 will be important, but so will be the phase 1 results from SAN711. That will allow us to focus on the indication that we actually want to pursue and how we want to move forward with that asset. The initiation of SAN903 will be very exciting in the second half of next year. And then on top of that, as I mentioned, we hired a new chief business officer, and the chief business officer, her sole focus will be on business development, which we all believe will truly unlock opportunities and potentially bridge many financial gaps that we have through non-dilutive raises. So I think there are plenty of milestones that we're excited about, and they will all be coming very soon.
Great. Well, thank you so much for answering the questions, and thanks again for your presentation, and we wish you all the best for your coming work.
Thank you so much, Michael. I appreciate it. Thank you, everybody, for listening.
And then we are ready for our next presentation, which will come from BioInvent International and CEO Martin Welschof. Welcome, Martin, and please go ahead.
Thank you very much, Cecilia. Happy to be here. So this is our forward-looking statement. As you know, we are listed on the Stockholm Stock Exchange, and I would like to start with a quick snapshot of the company. So we are a company focused on innovative immuno-oncology therapies with different mode of actions. We are pushing ahead with a portfolio. So we have currently 4 clinical programs ongoing, and by the end of the year, we will have a 5th program. We are a very integrated company, so we have a research engine, which comprises functional screening, and I will come back to that later in a minute. But also we have under one roof GMP manufacturing, which is a competitive advantage. The technology has now been validated through a number of collaborations. A couple of companies are listed here. Most recently, we did a collaboration with Pfizer, who picked targets and antibodies that we have selected with our platform. But also in the past, we did antibody discovery collaborations with Daiichi, Bayer, and Mitsubishi Tanabe. We're very proud about our strong institutional shareholder base. We have, as listed here, Redmile, Van Herk Investments, one of the long-standing loyal supporters, Omega, HBM, AP4, Merieux, Swedbank Robur, Invus, and Handelsbanken. We have a very solid cash position, so currently we have a runway until the end of 2024. And as I already mentioned, we're listed actually since 20 years on the Nasdaq OMX in Stockholm. So just very briefly to recap what has happened during 2021, was actually quite active year. So we have done a couple of clinical and preclinical milestones. We started very early during Q1 this year, presenting our data for our lead compound, BI-1206, where we saw first signs of efficacy. But then we also pushed aggressively ahead with our portfolio strategy. So we have started enrolling patients for our second and third compound. BI-1808 is the second one, and the third one is a collaboration that we have with Transgene, BT-001. And both studies are recruiting very well. We also had a very important preclinical milestone. So we have shown proof of concept data on our second anti-Fc gamma R2b program 1607 at the AACR this year. And then, of course, last but not least, we did a very successful financing during Q1, during which we raised close to $116 million US. Going forward for the rest of the year, so at the end of the year, we're planning to update the market on BI-1206 in both studies, in the non-Hodgkin lymphoma study as well as in the solid cancer study. And then, of course, another important milestone, as I already mentioned earlier, is our fifth program that will initiate clinical development by the end of the year, which is BI-1607. And there we plan to submit the clinical trial application by the end of the year. And as I already mentioned, I think earlier, we have a very strong focus on potential additional partnerships and milestones from collaborations that are already ongoing. So very briefly on our technology platform. So at the core, at the heart of the company, we have something which is called FIRST, which is really, as mentioned here, phenotypic discovery screening for new oncology targets and antibodies. And I will just focus on the unique features. So first of all, we are always starting from fresh human tumor tissue or patient material. And for that, we have established a very close collaboration with the local hospitals in Lund, such that we receive this material on a regular basis. And then once we have it, we do screening against our high-quality encoder antibody library and select specific antibodies against the cells of interest. And then before we really find out what those antibodies are binding to, we go through a number of animal models, so do a phenotypic screening and focus only on those antibodies who have shown strong therapeutic effects in a number of animal models. So that's the platform that, for instance, Pfizer has used to pick targets and antibodies of interest. And that's the platform that we have used to build our portfolio, which is summarized on the next slide. And before I go into further detail regarding the various programs, I'll just give you a quick summary and overview about the portfolio. And basically, we're focusing onto two different categories. One is around the target Fc gamma R2b, which is a very interesting receptor on the cells of the innate immune system. But we also have quite some focus on T regulatory cells from the tumor microenvironment, and there we're focusing specifically on two targets. One is CTLA-4 and the other one is TNF receptor 2. BI-1206, which is our lead program, has been already partnered with Ectin Pharmaceuticals for China, Hong Kong, Macau, and Taiwan. And that program is currently, or this antibody is currently in two different clinical development programs, one in combination with rituximab for non-Hodgkin lymphoma, and then the other one for solid cancer in combination with pembrolizumab. And I will go into details a little bit later. Then we have a second antibody called BI-1607. And that antibody is scheduled for the clinical trial application at the end of the year, and that will be our fifth clinical development program. Then very briefly, the second category where we focus on targets on the T regulatory cells from the tumor microenvironment. So there we have two targets. So the first one is CTLA-4. And there we have a collaboration with a company called Transgene. And there we combine our proprietary anti-CTLA-4 with their oncolytic virus platform. And this program has started recruiting by the beginning of the year and is recruiting very nicely. And then, as you can see down here, we're focusing on a target called TNF receptor 2, and this is one of the hot and upcoming IO targets. And there we have two different antibodies. The lead is BI-1808, and that program has also been started and is recruiting very nicely. And then we have a second program called BI-1910, and this is still in preclinical development. I should also mention that we have two clinical supply and collaboration agreements with Merck. One around BI-1206 in solid tumors in combination with pembrolizumab, obviously. And then we just recently signed a second contract for BI-1808, where we do single agent as well as combination with pembrolizumab. Now we'll tell you a little bit more in detail about the different programs. So starting with our most advanced program, which is BI-1206, which is in non-Hodgkin lymphoma in combination with rituximab. And there we presented actually first signs of efficacy at the beginning of this year. That was during Q1. And there we had 15 patients enrolled. Out of those 15 patients, 9 patients could be evaluated for response. And out of those 9 patients, we had 2 complete responses and 4 partial responses, which is a very good overall response rate, even though it's still early days. And more important, actually, that the complete responses have long durations, so it's a very high quality complete response. So in one case it was more than 1 year, and in the other case more than 2 years. So very encouraging first signs of efficacy. A little bit more detail on the indications that we're currently focusing on. So we're not focusing on all the non-Hodgkin lymphoma at the beginning. So working on follicular lymphoma, marginal zone lymphoma, and mantle cell lymphoma. And you see on this side the value driver. So we have a very compelling scientific rationale in anti-CD20 refractory B-cell lymphoma patients. We are positioning this as a chemo-free regimen since we have a favorable safety profile. And there's really a high unmet medical need for patients in second and third line. Also to mention that we have orphan drug designation for mantle cell lymphoma, which we got in 2019. On the next slide, very briefly, the study design. So as I mentioned, we are focusing on patients who have relapsed or are refractory and do not react anymore towards anti-CD20 antibodies, in this case rituximab. We are currently still in the dose escalation, so part 8, but we are at the end of it. So we're moving soon into dose expansion. And basically there are 3 work streams that we're currently focusing on, which are summarized on the next slide. So number 1, we are really trying to select the dose for part 2. And then we're preparing quite heavily end of phase 1 meeting with the FDA that we want to have as soon as possible. And then we're working very diligently with our partner in China, Ectin Pharmaceuticals, who are planning to get an IND approved for China at the end of this year. Coming to the second study of BI-1206, and this time it's focusing on solid cancer. So we are focusing on patients with solid tumors who have relapsed or are refractory against anti-PD-1 or anti-PD-L1. We are still in dose escalation, we are recruiting. And as I already mentioned earlier, so we are basically partnering there with Merck, which is a clinical supply and collaboration agreement, and will also provide an update towards the end of this year about the status of this study. So this slide gives a nice summary about the potentially broad application for 1206. So we're currently focusing in indolent non-Hodgkin lymphoma, and then in the solid cancer study on metastatic melanoma, as well as non-small cell lung cancer. If data allow and support, we can branch out into a broader non-Hodgkin lymphoma, but also other solid tumors. And I think what I also would like to mention at this stage is that there's also application potential in autoimmune diseases that we're not exploring ourselves, but through collaborations with different academic groups in order to establish in vitro proof of concept. Switching gears now, so going from anti-Fc gamma R2b antibodies to anti-CTLA4 and anti-TNF receptor 2 antibodies, which are expressed on T regulatory cells of the tumor microenvironment. And I will start talking about our collaboration with Transgene, which is around the compound BT-001. So BT-001 is basically our proprietary anti-CTLA-4 antibody, which is nicely differentiated from the one which is currently used in the clinic, ipilimumab. And this antibody genes we have cloned into the oncolytic virus of Transgene, such that when we transfect solid tumor cells, that when the virus is replicating, the virus is lysing the cells at the same time point, also producing anti-CTLA-4 at high concentrations. And what we achieve through that is that we have a very high concentration of anti-CTLA-4 in the solid tumor environment and a very low systemic exposure to the antibody, which of course is very important for the safety profile. This study has been started at the beginning of this year and is recruiting very well. And hopefully we can provide some updates around this study soon. Coming then to our TNF receptor 2 antibodies, and just to reiterate, TNF receptor 2 is one of the very interesting targets, probably a new immuno-oncology target, and we are at the forefront. So we are the company who is most advanced with a program against TNF receptor 2 since we're already in the clinic. And around that program we established our second clinical trial and collaboration agreement with Merck. Since we are not only testing BI-1808 as a single agent, as I will show you on the next slide, but also in combination with Keytruda. So this study is running very well. We have a very strong rationale for single agent as well as combination therapy. And we got an NDA approved by the FDA in April 2021. Just very briefly on the clinical study design. So as I already mentioned, we have two arms. So one single agent that has been already started. And then we also will explore combination with Keytruda, pembrolizumab. The single agent trial is well on its way. So that was initiated by the beginning of the year. Recruiting very well, very nice progression. And we are focusing there on 3 different indications. First, non-small cell lung cancer. But also ovarian cancer, and there's actually quite some interesting background to that. So when we did the screening on tumor tissue from patients, we worked a lot with ovarian cancer material, where we could see that TNF receptor 2 was highly upregulated on the T regulatory cells in the tumor microenvironment. And then we have a third indication. This is CTCL, which is a rare T-cell lymphoma, and that could provide potentially a shortcut to market. So in case we have strong data there, we could go there for conditional approval, because as for ovarian cancer, also for CTCL, there's a very high unmet medical need. Then a little bit later, we will start with the second arm, where we combine 1808 with Keytruda, and that will be focusing on non-small cell lung cancer and ovarian cancer. The single arm is already recruiting very well, and as I said, for BT-001, we also hope to have an update around that study in the near future. At the end, I would like to come back to the news flow for the rest of this year, 2021. So as I mentioned, so we'll provide an update around BI-1206, which is our lead compound for the non-Hodgkin lymphoma study, as well as for the solid cancer study. Then, of course, a very important milestone is that we push ahead with our portfolio. That means our 5th program will initiate clinical development, which is BI-1607, our second Fc gamma R2b antibody. And then we remain very active regarding business development. Thank you very much for your attention.
And thank you for the presentation. First of all, I would like to ask you what you would say is your biggest advantage if you compare to your competitors.
Yeah, there are actually a couple of advantages. So first of all, I mentioned already at the beginning the high level of integration. So in our company, we really have not only discovery, but also preclinical development, clinical development, and manufacturing. And that's a very strong competitive advantage because of timelines, basically. So when I started at BioInvent, we had 1 program in the clinic. That was August 2018. And now by the end of this year, we'll have 5 programs in the clinic. And you can do only this once you have this level of integration. The second thing, which goes, of course, hand in hand, is the science that we do. So we have a very strong immunologist team that can really work and find new mode of actions. And then thirdly, as I explained briefly, the discovery tool that we have first, which is a phenotypic functional screening tool. That is also, I think, the way we use it, unique for BioInvent.
And you mentioned here the fifth asset that you're about to enter a clinical study with. How will you keep the speed and the focus up in all your projects?
Well, obviously we have, with the most recent financing, we have grown the company. So a couple of months ago, we just had a core team in clinical development. Now we have a real team. So with clinical operations, regulatory, but also PK, PD, and of course program managers, etc., etc. So I think each program gets a proper resource so that we can really execute and make sure that we keep our timelines. And that's actually the main thing. So really have the right resources in play. And I think the shareholders that we got on board during Q1 this year, they also want us to build a diversified portfolio.
And finally, we'll end on a very general question. What is your strategy? How would you describe it regarding your clinical assets?
Yeah, so at the moment, I think our sweet spot is really to find new treatment modalities, new mode of actions. So that's, I think we're really good. And then translating this quickly into the clinic. So our current strategy is really afterwards, once we have shown proof of concept in the clinic, to find partners. And we have done so already for our lead asset with Ectin Pharmaceuticals. So, typically maybe we would start with a geography-focused partnership, keeping the rest of the world, either for ourselves or for bigger partners to come. And then eventually at some time point, so we have two indications, one for BI-1206 in non-Hodgkin lymphoma, where we could go for conditional approval. We could probably do the same for BI-1808 in CTCL. But that remains to be seen. That's of course data dependent. And in case we should see strong data there, then we also have the possibility to integrate us further forward along the process and value chain. But currently it's a strong focus on partnering.
But thank you so much for coming, and thank you for the answers.
Thank you, Cecilia.
And for the final presentation here from Lund, we are joined by RhoVac and CEO Anders Månsson. Welcome, Anders.
Thank you. So welcome to the RhoVac presentation. My name is Anders Månsson and I'm the CEO of the company. It gives me great pleasure to give this presentation today, especially as I can do this on the back of some really interesting results and achievements in the last few weeks, which has also been rewarded by an increase in the share price. But let's start from the beginning. What is RhoVac all about? Well, it's all about preventing cancer recurrence after curative intent therapy to the local tumour. Our lead drug candidate, RV001, is a Rho-C-based cancer vaccine targeting metastatic cancer cells, often remaining after primary tumour therapy, but coming in before the formation of metastatic tumours. The product is in phase 2B in prostate cancer. We're treating prostate cancer patients with biochemical recurrence with the ambition of preventing further progression of their cancer. For this indication, we have the FDA's Fast Track designation, and we also have financial backing from the European Innovation Council in the Horizon 2020 program. RhoVac is looking for a partner after phase 2B, which we deem will take place in the first half of next year. And we're looking for a partner capable of doing phase 3 development in this area and also eventually securing a global launch of the compound. Let's start with a patient. What is our mission? What kind of patient is it that we're trying to help here? Well, it's the patient that I think we all know. It's the patient who undergoes a cancer diagnosis, surely a tumultuous experience in anybody's life. But there is hope on offer from the physician because there are as yet no metastases. So a local therapy is recommended and performed. It could be surgery or radiation or a combination of the two. And this is done with initial success. But after that follows a period of intense waiting and anxious waiting on behalf of the patient. And we know, unfortunately, that a lot of these patients will experience a signal that suggests that they are at risk of progressing into metastatic disease. And this is what we want to prevent with our drug candidate, RV001, to prevent the progression of these patients into a metastatic state cancer obviously with a far worse prognosis. So let's examine the background, the scientific background of RhoC, which we use as an antigen. Already back in 2009, the American National Cancer Institute ranked RhoC as a prioritized cancer antigen, citing it was potentially ideal for cancer vaccine development. This was based on prior research suggesting that in normal cells, RhoC is silenced and has no interaction with the immune system, while as metastatic potential cells had an overexpression of RhoC, and these cells could be identified by the immune system, and these cells could be eliminated by the immune system. And this was also demonstrated preclinically in the diagram that you see on the slide. This is an effector target cell ratio diagram showing that metastatic cancer cells of different tissue origin could be eliminated by the activated T cells, the RhoC activated T cells. And the interesting thing in this diagram is to see that there is no major difference between the ability to lyse or eliminate these cells between the different tissue types of the cancer. So there's no difference here between colon cancer, melanoma, and breast cancer. The knowledge of RhoC and how it interacts with cancer has been increasing in recent years. If you look at the results in 2012, it was concluded that a majority of cancer types had an overexpression of RhoC in its metastatic cells. And in 2019, it was concluded that RhoC was indispensable in metastases. So our conclusion is that we have a potentially tissue-agnostic therapy concept that is aimed specifically at metastatic cancer cells, which is obviously of great interest. So what is the product concept of RV001? Well, it's a 12-injection cancer vaccination program initiated after primary tumor therapy and using RhoC-based antigen. RV001 is actually a 20 amino acid fragment of that protein. And RhoC is a protein that across tissue types is indispensable for the ability of metastatic cancer cells to migrate and spread and infiltrate other tissue, which is what makes them so lethal. And obviously the aim with our therapy is to prevent or delay cancer recurrence and progression by inducing a T-cell immunity to potentially remaining metastatic cancer cells based on their unique overexpression of RhoC. So we know that we have an antigen that can excite the T cells to search and destroy cells that have an overexpression of RhoC. But we still need to circumvent or elude another problem. Cold tumors has remained a significant problem in immunotherapy. We know that the tumor structure itself can give rise to defense mechanisms that exclude and eliminate the ability of T cells or other immune system cells to penetrate the tumor. So rather than fighting these tumor defense mechanisms, we target our therapy to be a non-toxic nip-it-in-the-bud therapy, targeting the deadly seed of potentially undetectable metastatic cancer cells, potentially remaining after primary tumor therapy. So this has moved on to clinical phase, and we've done a phase 1/2 trial back in 2018 with a 1-year follow-up in 2019. This is published information, and this is available through our homepage. I will read to you the main conclusions. We induced a potent and long-lasting T-cell immunity in the majority of these patients. The majority was here 86%, so quite a vast majority. We had excellent safety and tolerability, and this was confirmed also in the midterm analysis of our phase 2 data, which I will talk about later. And vaccination against RhoC could potentially delay or prevent tumor recurrence and metastasis formation, and this is based on the analysis of PSA values. Recently, we also presented the headline data from a 3-year follow-up of the phase 1/2. So in this 3-year follow-up, we found that no patient had progressed to metastatic state and no patient needed other therapy. And all but 3 patients had completely undetectable PSA levels, and patients that had a weak PSA signal after 3 years all had a prolonged PSA doubling time, indicating a slowdown of their disease progression. And after 3 years, only one single patient who was a responder after the 1-year follow-up was now considered a non-responder. So this patient population was almost in its entirety intact as regards its T-cell immunity. So obviously these very promising results had to be tested in a larger setting and with a placebo control group. And that is what we've done in our phase 2b study, which started in 2019. Here we have recruited 180 patients across 35 centers in Europe and the US. The aim of this study is to show that we're able to prevent progression, which is defined as PSA doubling, clinical recurrence or death in prostate cancer patients with biochemical recurrence after curative intent therapy. Recruitment is finalized and we anticipate to be able to conclude the study and compute results already in the first half of 2022. So why is this so interesting commercially? Well, if we start by focusing on prostate cancer, let's examine this diagram. If you look at the diagram, you'll see on the left side a grey bar indicating the number of patients diagnosed each year with local prostate cancer in the 7 major markets of the world. If you go to the right side to the orange bar, you will see the number of cases of metastatic cancer being diagnosed each year in the 7 major markets. Now, if you know something about prostate cancer, you'll know that most of the therapy in this area is hormonal therapy, which is by necessity focused on the metastatic cancer patients because of the side effects. But because we have designed our therapy as a non-toxic therapy, a vaccination therapy, we can aim earlier, we can treat earlier stage patients before they have symptoms and before they have metastases. Why cannot the currently available products do that? Well, it's because of the nature of these hormonal treatments. They're called ADT or androgen deprivation therapy, and they have castration or castration-like side effects. In the acute phase, those side effects can often lead to erectile dysfunction or loss of libido, but in the long term there is also metabolic side effects or metabolic risk, especially in the cardiovascular area. But our treatment is potentially possible to use as a treatment for earlier stage patients, and obviously these patients are several times more plentiful than the patients currently treated by the entire prostate cancer drug market today. This is why this is so commercially interesting. However, it doesn't stop there. Our first clinical proof of concept targeted is the indication currently pursued in the phase 2b program, which is preventing progression and clinical prostate cancer recurrence in BCR patients. However, we could potentially expand from there, first within prostate cancer. We can possibly go to earlier stage patients, even further earlier stage patients, as a replacement for surgery or radiation in patients that are not suitable candidates for these procedures, or in patients with other risk factors than a rising PSA. We could also go for later stage patients, combining with ADT in patients that present with metastases. And the rationale for this is that, well, ADT treats the hormone-sensitive cancer cells, whereas each prostate cancer also has a number of cells that are not hormone sensitive. And of course, our treatment, being a vaccination, doesn't discriminate between hormone sensitive cells and non-hormone sensitive cells. So adding it on top of ADT might actually prolong the lifespan of these patients, or at least add to the effectiveness of the entire treatment. But as I've made a point of the fact that RhoC is completely tissue agnostic, the really big expansion possibilities would also be in expanding into other cancers. Recently, Edison Group has performed a new and updated analysis of RhoVac, and they have the following to say about some key parameters. They forecast peak sales of our product, given successful development, of course, of $1.8 billion. In fairness, I should say that other analysts have other forecasts around $1 billion, but at least it's blockbuster potential. This means also that the value of RhoVac is adjusted upwards to 1.6 billion, corresponding to an 85 krona per share price. And it should be noticed that this valuation considers the currently pursued prostate cancer indication only. If we're able to expand within prostate cancer, if we're able to expand outside of prostate cancer, this valuation would of course increase. And also it's explicitly stated in the analysis by Edison that if we have a positive readout of phase 2b, then the valuation would more than double. So the current situation of RhoVac is the following. We have ticked the boxes of a great many milestones. Ahead of us we have essentially only a few. The first half of next year we deem to be able to conclude the phase 2b study and compute the results. And after that we will pursue a deal with a large oncology, possibly urology focused company that is able to carry out the phase 3 stage development and also a global launch, making this product available to patients all over the world. So I will try to summarize with this slide my entire presentation using only 3 symbolic images. The first image is obviously the picture of a rose, which symbolizes RhoC. Please remember that RhoC overexpression is a unique trait of metastatic cancer cells, but it is not unique to any particular type of tissue or cancer. And therefore, this precipitates a hope that we have a tissue-agnostic cancer vaccination concept on our hands. The second image is a picture of buds, and this symbolizes our nip-it-in-the-bud approach or preventive approach that we suggest for RV001. So in suggesting this, we avoid trying to treat possibly immune-excluded and therapy-resistant tumours, but we suggest that this approach of treating after primary tumour therapy, but before the formation of these metastatic tumours, is the right approach. And the final image is the image of a racetrack. Obviously, it's a very fast track, and that's a play on words with the fast track designation that we have from the FDA. And it's also, of course, a symbol of a fast track to the future for RhoVac as it stands currently. We deem able to produce the results of the phase 2b study already in the first half of next year. With that, I thank you very much for your attention, and I leave you with some contact details. Thank you very much.
And thank you for a very interesting presentation.
Thank you.
I have a couple of follow-up questions. You are in the middle of a rather intense news flow at the moment, and you are focusing more and more on, like you said, reaching a deal for your lead candidate. Can you describe the situation that RhoVac is in right now?
Yes, it's a very happy situation. I mean, we are very happy to have taken us through the challenge of recruiting patients for a clinical trial in prostate cancer in the midst of the pandemic. But now we have full recruitment, and of course that feels great. It's a great achievement. We're also happy that we have recently produced very positive phase 1-2 results in terms of our 3-year follow-up. And those results actually hold great promise for the future and for the readout of the phase 2b. Then we are, of course, happy that we are not so far away from having the conclusion of the phase 2b, so that is deemed to happen in the first half of next year. And in anticipation of that, we've also brought Stifel Investment back on board. So we are able to say that we have the best deal team on board to secure the success, provided that we have a good readout, of course. And I'm also very happy to conclude that we have an increased valuation both from third-party analysts and in the actual share price. So it's good and exciting times in RhoVac.
I understand that. Like you said, you've brought Stifel Investment Bank on. Can you tell us a little bit more about that?
Yeah, I mean, we're a small biotech company, but we have a potentially really, really big opportunity on our hands. So, of course, we need to invest in having the best possible team in place to secure a good deal for RhoVac and for the continued development of this drug.
And finally, of course, the readout from the phase 2b study next year is your next big milestone. But what activities will you be focusing on until then?
Well, we do market research in other cancer indications. As I just presented, of course, we do have the opportunity to have this compound go into other cancer indications as well. We are not going to do it in RhoVac apart from preclinical development, but we will be able to suggest to our partners what to prioritize and where to go. Obviously, we're also updating and taking care of our data room, so that's ready for the readout. And then we also have other development that we're doing in terms of preclinical and formulation development. So all this needs to come to conclusion fairly soon, so that we're able to have everything ready by the time we're ready to negotiate.
So you certainly have busy times ahead of you then.
Yes.
But thank you so much for taking the time and telling us about it.
Thank you so much.
Thank you.
That concludes the fall edition of the BioStock Life Science Summit 2021. All the presentations you have been watching can be seen again and again on our website and on our YouTube channel. I'd like to extend a big thank you to all of you for watching at home and to our event partners Nasdaq and Potter Clarkson. Looking forward to seeing you again soon. Bye.